our YouTube channel of APP Online Convention. It has been connected, and now the participants are also joined. And
so we have 3 chairpersons, I think. Professor Goel, Professor Anju
yes,
Goel is there.
Professor Anju Goel is there, Dr. Ramesh is there.
And Professor Dinda is there. So,
yeah.
And we have our first speaker, Dr. Kamini, she is there. Okay. And second speaker is there. And I think last speaker, 2 speakers are not, but we can continue the session. I will contact them and join.
Yeah.
Thank you.
I think we can start. Dr. Gurpreet, you have joined with 2 links, I think.
Yes, sir. I'm switching off that.
Yeah. Please mute the other system so that it will not be disturbing.
Yes.
Welcome all of you for the 5th scientific session of APP, Association of Pharmacy Professionals, 9th annual convention. This is the 9th year after the conception. 2011 it has been started, and the association has now more than 1250 members, and we are celebrating, and the first virtual international convention of APPA, and we are very happy to organize this online. We welcome all the respected chairpersons and eminent speakers for this session. Welcome all of you on behalf of the organizing committee, the Association of Pharmacy Professionals, Rajasthan State Branch, and the Central Branch, as well as the Sunrise University, the host organization. Welcome all of you, and not taking much time, I would like to call upon the speakers. First speaker of this session is Dr. Kamini Govinder from University of KwaZulu-Natal, Durban, South Africa. She is going to talk on sub or supercritical fluid chromatography purification of biologics. So one of my organizing team colleague will recite the profile and invite for the talk. Please.
Thank you, sir.
But now I think Dr. Kamini has not connected by this time, I think.
Okay.
But I have seen once, I think so.
Yeah, she connected, and now after.
I think maybe some problem with the
Yeah.
connection.
Okay.
Can we go with the...
Yeah. You can go with the second speaker, and then she can be on his or her place.
Yeah. Now I request the organizing committee member to call upon Dr. Vettiselvan Subramanian from the Masa University, Malaysia, to talk on the topic translational animal research in drug development. So he'll be delivering his first lecture in this session. So please recite his profile and invite him to talk.
Thank you, sir.
Yeah.
I would like to introduce a brief profile. Dr. Vetri Subramaniam. Currently, he serves as Associate Professor of Pharmacology, Faculty of Medicine, Nursing and Bioscience, MAHS University. He did his graduation from Annamalai University. He has more than 10-year experience in teaching and research. His research focuses on metabolic diseases and alternative treatment approaches. He published more than 35 research review articles in reputed journals. He is a member of various professional associations, that is Indian Pharmacological Society, APP, Malaysian Scientific Association, IACP, and Malaysian Society of Pharmacology and Physiology. Currently holds the position of the executive member of Indian Pharmacological Society and Vice President of APP Malaysia. I welcome you, sir, and share your STEM research experience to translational animal research in drug development. Over to you, sir, please. Sir, unmute yourself.
Sir, you are not audible.
He
No,
has to
sir.
share his presentation.
No, actually, he has to reconnect his audio through computer audio. Are you hearing us, sir?
Hello.
Yes, sir. Yes, sir. There is small technical glitch.
I think Dr. Kamini is there. If you want to ask.
Yeah. Yeah, we request then Dr. Gurpreet, please recite the profile of Dr. Kamini Govinder and please to start. I request Dr. Vitiselvan, please check your audio connectivity because your voice is not reaching us.
Okay, can you hear me?
Yeah.
Yeah, Dr. Govinder, we can hear you.
Thank you. Okay, thank you.
Yeah, Dr. Gurpreet Singh, are you hearing us?
Sharing.
Yes, sir.
Yes.
Shall I recite the profile of Dr. Kamini?
Yeah, please.
Okay. Good morning, everyone. I, Dr. Gurpreet Singh, on behalf of the organizing committee, welcome you in the 5th session of this second day of virtual 9th Annual International Convention of Association of Pharmacy Professionals organized by Faculty of Pharmaceutical Sciences, Sunrise University, based on theme Recent Advances in Pharmaceutical Sciences, Potential Role of Herbs and Health Challenges. It's my pleasure to recite the brief bio sketch of Dr. Kamini Govinder. Dr. Kamini Govinder presently enrolled at the University of KwaZulu-Natal in South Africa. She completed her UG, PG and PhD in biochemistry from the School of Life Sciences, University of KwaZulu-Natal in South Africa in 2017. Her PhD study resulted in 3 international publications and was also presented in 1 international conference named the 7th European Federation of Medicinal Chemists Young Medicinal Chemists Symposium and a national conference named the Royal Society of Chemistry South African Chemical Institute Young Chemists Symposium 2020, where she won the best poster in the state. Her PhD study can be employed as a key platform for supercritical fluid chromatography purification of biologicals in the pharmaceutical industry. In future, Dr. Kamini aspires to be the world-renowned scientist. So I would like to invite Dr. Kamini Govinder for the first lecture on title Supercritical Fluid Chromatography Purification of Biologics. Over to you, ma'am.
Okay, thank you. Can you see my slides or is it?
We can see.
Okay, thank you. Good morning to everyone. Thank you for giving me this opportunity to present at this convention. Thank you to the APP and the president and all chairpersons and everyone present here today. It's a good opportunity because you get exposure to everyone across the globe. So I just finished my PhD in pharmaceutical chemistry. My name is Dr. Kamini Govinder, and I'm from the University of KwaZulu-Natal in Durban, South Africa. I come from the department, the Catalysis and the Peptide Research Unit in College of Health Sciences in South Africa. So my talk for today is based on the sub-supercritical fluid chromatography of biologics. Biologics are imperative in the pharmaceutical industry because it's used as active pharmaceutical ingredients. And yeah, so then but biologics, so my talk will be based on the biologics pertaining to diabetes. And diabetes is one of the most prevalent non-communicable diseases worldwide. So approximately 463 million adults suffered from diabetes globally. This number is projected to increase to approximately 700 million in 2045. So this number creates a huge burden on current insulin manufacturers. Therefore, we need to create a better, more affordable way of providing insulin to people across the globe. The current method of insulin purification is HPLC. However, high-pressure liquid chromatography is expensive and it uses large amounts of toxic organic solvents. So in our study that I conducted in my PhD, so what I did was I used cutting-edge SFC chromatography to purify the human insulin. So figure 1 just shows you a CO2 phase diagram. So supercritical fluid chromatography uses a condensed gas, such as carbon dioxide, to purify, which is used as a mobile phase to purify our biologics. So it has to be at a critical pressure and a temperature which is above 31 degrees Celsius and 74 bar. So a temperature, so this, then it'll be regarded as a supercritical fluid. So the aims of my study for my PhD was firstly to biosynthesize the human insulin using E. coli as a protein expression host via recombinant DNA technology. Thereafter, to purify the human insulin using SFC chromatography, I detected and quantified the aforementioned protein using
Maybe some interruption happen.
I think Madam has lost the connection.
Yeah.
I think in South Africa connectivity issues are there. So we were worried about in India.
Yeah, definitely. But it happens there also. Surprise.
Yeah, she's there.
Okay.
sorry.
It happens.
Is it
Share
still,
screen.
is it?
Yeah. Range.
Sorry about that.
Yeah,
I think I'm having
yeah, you can share.
issues.
Wait, don't press enable editing. Start slideshow.
Yes. Okay. Okay. Can you, okay, so is it fine now? Sorry. I think I lost connection.
No problem. Proceed. It's all set now.
Okay, yeah. So basically thereafter I went about, I did a PCR cloning strategy. And so firstly, we identified the gene of interest, which was insulin. And we used PCR, polymerase chain reaction, and we amplified our gene of, I amplified the gene of interest. And thereafter, I did a digestion of the PCR products, followed by ligation to the respective plasmid, and then transformation in step 4. And then what you do is you isolate your plasmid So you do a plasmid isolation and followed by construct verification. Thereafter, I verified my positive transformants via PCR, isolated the plasmid and transformed into my expression host. The verification of the positive transformants were conducted via PCR as well. And subsequently, I optimized the expression of the protein using various IPTG concentrations, 1 millimolar, 0.5 millimolar, and 0.1 millimolar, respectively. Thereafter, I went about to detect the protein to see if it was actually insulin or what protein was in that I overexpressed in the host cell proteins, because there's a lot of host cell proteins that you can get. And I verified this using LCMS and MALDI-TOF-MS. Thereafter, I went about purifying the biosynthesized human insulin that I just biosynthesized using SFC chromatography. This was followed by lyophilization, the recovery of the biosynthesized insulin, protein sequencing, and I used MTT assay, which was conducted in vitro. Thereafter, we tested solubilities of 3 peptides, a tetrapeptide, an octapeptide, and a nonapeptide, because the insulin, when we purified it on the SFC, we had a lot of phase separation and a lot of issues. So we wanted to just investigate further using methanol only, because insulin, when we dissolved it, we initially used water on the SFC. So we just wanted to investigate further with other peptides other than insulin. And so we conducted SFC purification of the aforementioned peptides. And so thereafter my PhD, I've got the following papers. So my paper 1 was based on a novel and more efficient biosynthesis approach for human insulin production in Escherichia coli. So this was based on the development of a novel and more efficient method of human insulin in E. coli. So the biosynthesized human insulin was verified via protein sequencing, and the biological activity was assessed. So in figure 4, it's a peptide spectrum, which shows that the protein sequence of the crude biosynthesized human insulin was in fact, it matched the human insulin from the protein database with 100% probability. So therefore, I verified that it was in fact human insulin. And in figure 5, it's a graph illustrating the cell viabilities of HepG2 cells under hypoglycemic conditions for the standard and the crude biosynthesized human insulin. And this was conducted at 3 concentrations, 50 millimolar, 100, and 150 millimolar, using HepG2 cells, which is liver cells. And what this graph basically shows is that the standard and the biosynthesized human insulin displayed a similar degree of efficacy to each other. So the biosynthesized human insulin, which
turn off.
Sorry, can you hear me? Okay. It's fine. And thereafter, I went to purify the biosynthesized human insulin using sub-supercritical fluid chromatography. And this was my paper. So it was based on the purification of human insulin, whereby various columns were assessed. And LCMS and protein sequencing were used for detection and quantification respectively. Thereafter, the biological activities were assessed using an MTT assay. Figure 6 shows the SFC chromatograms of the standard sample of human insulin. So the standard human insulin was used to assess which columns will be best suited to purify the biosynthesized human insulin. So in figure 6A, I used a silica column, but however, the silica column did not give proper separation, and figure 6B was an ethyl pyridine column. There too, the separation was not too good. And figure 6C was a diol hylic, and D was a PFP. Out of the 4 columns that were tested, the PFP gave the best separation. Therefore, we proceeded to purify the biosynthesized human insulin using the PFP column. And figure 7 is a peptide spectrum of the biosynthesized sample of insulin after SFC purification in the peptide sequence to human. So this gave a 96% probability to that of human insulin on the database. So after SFC purification, this shows that the sample, the biosynthesized human insulin was retained. It did give the same sequence as that of the standard. So we went about testing its biological activity after SFC purification under normal glycemic conditions and hypoglycemic conditions. And this graph, figure 8, basically shows that the activity of the biosynthesized human insulin was retained after SFC purification. So that, and it was also conducted using HepG2 cell lines under 50 millimolar, 100 millimolar, and 150, sorry about that, concentrations. And thereafter, I went about to study so I had to develop a method because we had problems with phase separation with the water that was obtained in insulin sample. So we tried to solubilize peptides, 3 peptides, a tetrapeptide, octapeptide, and a nonapeptide. And so the peptides are highly versatile in nature with several medicinal benefits such as antiviral, anti-cancer, and anti-diabetic properties. So we developed this method using SFC chromatography for the tetrapeptide, octapeptide, and nonapeptide. And in table 1, you can see the molecular weights and the sequences of the respective peptides in the study that was used. So my tetrapeptide was an insulin beta chain subunit, which was 15 to 18 the residues. And the octapeptide that was used was angiotensin 2. And the nonapeptide was an insulin beta chain peptide with 15 to 23 the residues. So paper 3 was based on the SFC purification of the aforementioned peptides, and they were tested on various columns such as the PFP, a diol hylic, and a 2-ethylpyridine column. The 2-ethylpyridine column was selected for further analysis because it gave the best separation and best peak shapes and good retention times. So the recovery tests were conducted on these columns. So figure 9 basically illustrates the SFC chromatograms that were obtained for the separation of the tetrapeptide. So this was done with the 5 to 60% gradient. And in 9A, it was a PFP column that was employed, and the baseline noise was not too good, so we didn't proceed with that column, and there wasn't 100% separation on that column. And then 9B had a diol hylic, and C and D were ethylpyridine. The ethylpyridine column gave the best separation because we could separate the impurities from the pure, the impurities and the pure compound. So thereafter, we went about to do an isocratic mode, so a 20% isocratic for the tetrapeptide on an ethylpyridine column. And so this was figure 10, and you can see that there's a separation. So the higher peak, the bigger peak is actually your product of interest, and the smaller one is your impurity that was separated from the compound. Thereafter, I went about to separate the octapeptide. The gradient worked fine with the octapeptide. So figure A, 11A is the PFP column. You didn't get good separation. The baseline noise was not too good as well. So then we didn't proceed with that column. And so B was a diol hylic, and you have C and D, as was the ethylpyridine column, which gave good separation. And so we proceeded to do a separation of a nonapeptide, which is also conducted on the same columns. And as you can see, in 12A, the PFP column also had a significant baseline noise, and B, with the diol Hylake, had the peaks were too close, so we couldn't proceed with that column. And then we went about to proceed further to separate this nonapeptide on an ethylpyridine column, and we obtained the best separation, as you can see. So table 2 represents the recoveries of the tetrapeptide, octapeptide, and the respective nonapeptide post SFC purification. And we obtained 80% recovery for the tetrapeptide. We had 100% recovery for the angiotensin 2, and we had 102% recovery for the nonapeptide. So in future, what we'd like to do is we can focus on the SFC purification of other essential biologics in the pharmaceutical industry. Yesterday, there was a talk on how biologics is very important in the pharmaceutical industry as they're using monoclonal antibodies, recombinant proteins, and these are all going to be used for treatment of various diseases, and especially now we have the COVID around, so we need more purification methods other than the current ones because they're very expensive. So I'd like to acknowledge the following people from my department in South Africa. My supervisors, Professor T. Govender, Professor Kruger, Professor Naika, and my research colleagues as well from the Catalysis and Peptide Research Unit. Thank you all for your support throughout the study. And I would also like to thank Professor Nirmala Gopal for giving me an opportunity to present at the APP conference. And I'd also thank you so much for your attention. And I'd like to thank the APP, obviously, for inviting me to speak here today. And sorry about the technical difficulties. And yeah, thank you.
Thank you, Dr. Kamini. And now we call upon to the next speaker, Dr. Vetriselvan Subramanian, and I request my colleague to recite the profile of Dr. Vetriselvan.
Sir, excuse me, can you hear me?
Yes, sir. Yes, sir.
Okay, sir. Thank you.
Yeah, you may proceed. Yeah, Dr. Gurpreet. Profile is already recited, sir.
Okay. Sorry.
Are you hearing me, Gurpreet?
Yes, sir.
Sir, I can hear you.
I'm
Yes,
hearing
sir.
you, sir. Shall I recite, sir?
Yeah. You may please start, Dr. Vetriselvan, sir.
Sir, shall I display my slides?
Yeah, you can
You are joining
display.
the new device.
Yeah, I joined new device, sir, because that the computer is not access.
Okay.
Okay. Then now you can present your slideshow.
Yeah, just a minute. Sorry for the interruption. Just wait, sir.
You can share your slide.
Yeah, just a minute, sir. I'm trying.
think Dr. Daya sir, Dr. Rohit Kumar Verma has not yet joined.
I asked, sir. I will again inquire about this.
Yeah. Yes, sir, please. This is for your knowledge.
Professor Jayapalan, we can go ahead with our next speaker.
Yeah, he's trying, I think. Sir,
Sir, I am trying just because.
Okay,
the
Dr. Sullivan, you can solve we will give chance to the next speaker, so then you can come.
Okay.
yeah, but then we call upon the next speaker, Dr. Kamal Dua, University of Technology, Sydney, Australia, is going to talk on the topic exploring cellular and molecular mechanisms of the phytoconstituents encapsulated drug delivery system. So we may move on to the next speaker. If Dr. Kamal Dua is ready hearing us, sir, please unmute your mic and camera, and you may start your presentation, sir.
Shall I recite the profile of Dr. Kamal Dua?
Yeah.
Dua sir, आपका lecture announce हो गया.
Okay, I'm just going in. Okay. 2 minutes. I'll just join.
Yeah, thank you, sir. Here I'm going to recite the brief profile of Dr. Kamal Dua. Dr. Dua is a full-time lecturer based at the discipline of pharmacy, Graduate School of Health, GSH, the University of Technology, Sydney, Australia. Dr. Dua holds dual PhD in pharmaceutical science and in immunology and microbiology. Dr. Dua published 123 articles, 6 book chapters, and 2 books. His 5 best publications highlight his expertise in biological studies, including cell culture and experimental models of chronic inflammatory disease and pharmaceutics and drug delivery. Dr. Dua won 6 project grants, including from the Department of Industry Innovation and Science, UTS, IMU, Malaysia, both as chief and associate investigator. Dr. Dua established his research program on influenza infection in respiratory diseases. Dr. Dua is an active member of research committees in GSH, including the Board of Studies, Discipline of Pharmacy Curriculum Development and Research. He is a member of the UTS Academic Board Research Committee and the interview panel for recruiting GMP program staff as well as students. He has supervised 6 PhD students, 14 masters, and 28 undergraduate students, and some of are in the pipeline also as well. Dr. Dua has reviewed 12 PhD, 10 master theses for national and international universities. He has peer-reviewed manuscripts for more than 15 international leading journals, including Molecular and Cellular Biochemistry, European Journal of Pharmacology, and many other reputed journals. Dr. Dua is also on the panel of editorial board for various journals, including Bantam Science and Springer Nature. Now, I would like to invite Dr. Kamal Dua for delivering his lecture on topic exploring cellular and molecular mechanisms of the phytoconstituents encapsulated drug delivery system. Thank you, sir. Over to you, sir. Sir, please unmute yourself.
Thank you. Sorry, just give me a minute.
Yeah, sure, sir.
Okay, can everyone see my slides, please?
Yes,
if
sir.
you can confirm.
Yes, sir.
Okay,
Make it
great.
visualize.
Okay, so is it clear? Is it a full screen mode?
Yes, sir. All done.
All right, thank you so much. And I'm really sorry for being a bit flustered because we got some guests at house and we've been running around like a chook. So just excuse me for that. So I'm going to talk about the potential of actually exploring quite a number of phytoconstituents which are encapsulated in the drug delivery, like nanoparticles, liposomes, and this is what my PhD students and lab is working towards. In terms of the research area, my lab works on the respiratory diseases, but not restricted just to the respiratory diseases. We primarily works in quite a number of inflammatory diseases. So as we know, in one of the latest paper, which was published in Lancet, and that has clearly shown that how intensely the respiratory diseases are actually affecting the Indian population. And it is prevalent across all the regions of the India. And if we really look the red color regions on the graph, and that shows in the northern part of the India, they are extremely highly prevalent as compared to the south part of India. And every day, off and on, you can see that there are a number of news coming out. This was just one of our news at Word, clearly showing that World Asthma Day, India chokes, and the sales of the medicine has rise from 43% in 4 years. And this is what all is happening is a prime reason is a pollution. And the people in the India also, they quite a lot of the people smoke. And because of the smoking, one of the common respiratory disease is the chronic obstructive pulmonary disease. So considering those important problems, so my lab focuses on two important diseases. In terms of the respiratory diseases, we focus on the asthma, COPD, and the lung diseases, particularly in the lung cancer. So if we look at the respiratory burden, which is giving these diseases on the healthcare system and economy, in asthma, almost according to the World Health Organization, 300 million people worldwide, they are affected with asthma. And if we look in India alone, almost 18 million people, they are being asthmatic, and that itself cost around 2.7 billion of Australian dollars. Likewise, the other disease which I was talking about is the chronic obstructive pulmonary disease. It is because of the smoking, and it is also known as emphysema. Almost 64 million people globally are affected by this disease. If we look at stats in India, almost 22 million people out of that 64 is just from India. And it cost us around 1.1 billion of the Australian dollars. If we look in different infections, including the influenza infection on which I work significantly, we have seasonal pandemics every year. And in 1918, we have a big flu season, like now it's COVID. So in a like way, we were having a flu infection, which has almost cost around like deaths of 250 to 500,000 deaths worldwide. In India, almost 43% of episodes are always of the airway infections, and out of which 14 to 12% are just because of the flu. And now what we are encountering is all around COVID infection. And the world has just been trying to find the treatments. UK has just announced that they got a vaccine, while they need to really prove the results yet. They are not yet out whether the patients are getting those antibodies well developed or not. So that clearly shows there is a high impact and a tendency to work in an area of respiratory diseases. And that leads to the importance of the drug delivery, because you are targeting the inflammation in lungs. And that's really difficult because you need to get a patient compliance and you need a suitable drug delivery pharmaceuticals. If you really look at the structure of our respiratory system, the way you go down from the trachea to the terminal bronchioles, the size will completely reduce. And if you look, the size is the reducing, and that reduction in the size, it means your drug delivery, the way you are being giving through, it need to reach those particular areas of those small zones. This was one of the book we recently published with Elsevier, and that has containing a numerous descriptions of the drug delivery systems we are targeting in the respiratory diseases. The most important thing what we need, it should be patient compliant. It should be effective. It should have a therapeutic value. It should have a lesser side effects, and it should be a better therapeutic potency. So what are the things we normally consider when we design a respiratory drug delivery? So all the physicochemical parameters, I'm not going to talk about these basic things because anyone can read through the books. We can also be considering the various factors which are important in terms of the devices we are using to deliver those drugs. And finally, the pharmacokinetics and the dynamics once your drug actually reach into the respiratory system. Now, what we do particularly here. So I'm based at a University of Technology, Sydney, which was being mentioned in the introduction. But my research group actually is based and split it between two different parts. So I am a part of a Center of Inflammation in the Centenary Institute, which is a part of Uni Sydney as well as the UTS. So we try to focus on the drug delivery system, which can show the release of the drug in a controlled release. And we use all different type of nanoparticles, including the modified nanoparticles like liquid crystalline, solid lipid, liposomes. And with this, we normally do this in association with a certain number of people from the UNSW, University of New South Wales, Ulster University in UK. We got a Free State University in South Africa, University of Witwatersrand in South Africa, and few collaborations from India from MDU, Manipal College of Pharmaceutical Sciences, NIPER, and LPU. You might have seen there quite a number of papers we got in respiratory diseases with these collaborators. What we target, we target all the important biological moieties we have identified in the experiments, whether they are microRNAs, which we see they are upregulated in the diseases, or they can be even the cytokines. And the most important particular thing we focus in our group is we target on the indigenous medicinal plants. There are various plants which are having a potential therapeutic activities, and we are using those to get into a novel drug delivery system like liposomes or the solid lipid nanoparticles, and then we are targeting into the respiratory diseases. And for this, I have collaborated with the National Institute of Complementary Medicine, which is a part of the Western Sydney University, as well as the Queensland University of Technology with Trudy Collett. We also easily get the patient samples. So I got a collaboration with Professor Peter Walk from the John Hunter Hospital in the University of Newcastle, where I'm based as a conjoint lecturer. So these are some of the papers we have published. So we have shown with liposomes, as well as with the different type of the nanoparticles, including liquid crystalline, solid lipid nanoparticles. And you can see more or less the drugs we are targeting, they are the natural based origin, such as curcumin. We have targeted quercetin, celestrol. We have even targeted recently rutin. And today I'm going to show you a bit of a data of my current study, rutin, which we have just published in Toxicology in Vitro. And also the tuberculosis, we have been targeting the various synthetic-based drugs like rifampicin, isoniazid, and pyrazinamide. On top of that, currently we are working on various other drugs like boswellic acid, narogenin, lensclunaris, and the berberine. Now, there are quite a significant number of the review articles we have published in this area in quite a number of reputed journals as well. And as I mentioned, so the research always which we carried out is a part of a different collaborative institutions where I'm based in Australia. So HMRI, which is the Hunter Medical Research Institute, is a part of the University of Newcastle and the John Hunter Hospital. So we get clinical samples from there. All the in vitro work and the drug delivery work is being done in a UTS as well as done in the Centenary Institute which is a part, as I mentioned, of the University of Sydney, as well as the UTS, where I'm based at and my group is based at. So what I'm going to do now is I'm going to show you some of the data of one of our study. Just give me a minute. I don't know why it's not showing. Okay, so I'm going to talk about one of our current study, which is actually about to get submitted for the publication, about the liquid crystalline nanoparticles we have used to encapsulate rutin, and we have targeted the non-small cell lung cancer in the proliferation and migration. So as we know that the cancer is one of the leading cause of the problem around the globe. And if we really look at the figures in terms of the statistics, it clearly shows that lung cancer is one of the 5th most common cause diagnosed and one of the leading cause of deaths in Australia. In 2018, almost, it is estimated that close to more than 9,000 deaths were attributed to the actually lung cancer. And the 5-year survival rate of the lung cancer is just less than 14%. Now, this clearly shows that there is a need of research in this area to target lung cancer. And to do that, the major risk factors, what we have identified in this disease includes the tobacco smoke. There might be a genetic makeup in the patient. COPD, the smoking is one of the major cause of the cancer. Particularly lung cancer, and the various environmental factors, as well as the chemicals such as asbestos. Now, and as I mentioned earlier, tobacco smoke almost equivalent to 85% of this actually lead to the lung cancer. So you can imagine the cigarette smoking is one of the major contributing factor in the pathophysiology of this disease. Now, oxidative stress has really been playing a significant role. In this particular study, we have 2 different axes. So in my previous presentation, I have shown that we recently published showing how oxidative stress is actually playing a role in lung cancer. And the second paper is purely on the migration and proliferation using the A549 cell lines in lung cancer. So as we know that in a cigarette smoking, when you smoke, automatically the epithelial cell and the macrophages, they really get activated in the lung cells. And because of that, it impacts and leave and attribute quite a significant effect on the increase in the oxidative stress by the oxygen, by the reactive oxygen species. So what it will happen is, because of this, there is an imbalance between the reactive oxygen species and the antioxidants. And because of that, it started building up the oxidative stress. Now, because of this oxidative stress, it targets certain number of cells. That includes the epithelial cells, and they even identify the macrophages. They start recruiting quite a number of the inflammatory cells and finally lead to the disease by 3 different mechanisms, with the protein damage, membrane lipid damage, and the DNA damage. Exactly on these grounds, what we have done, we have used this rutin as one of the compound, which is a flavonoid and can be found in ample number of the food products, including wine. It has clearly shown in the literature, it has various ample biological properties, including anti-cancer, antimicrobial, anti-inflammatory, antioxidant, antimicrobial, and anti-hyperlipidemic, and even platelet aggregation inhibitors can be used, rutin. Now, what we have done, we have prepared the liquid crystalline nanoparticles using 2 different polymers, monolin and polexamer 407. And these particular nanoparticles we evaluated for the basic, which every single lab around the globe do, the basic physical-chemical characterization for the particle size, zeta potential, and the PDI, all those basic things, including some of the drug compatibility studies. Then we have actually looked into the drug release profile, which is in vitro drug release. And then finally, we have done certain antioxidant and the anti-cancer potential of these cells, of these formulations. So what we have seen in terms of the physicochemical characterization, it has really shown all the optimized and the standard values, what everyone observed, clearly showing that the formulations were stable and they can be used very effectively as a standard formulation in terms of the liquid crystalline nanoparticles. They were quite spherical in shape as well. So when we looked at the in vitro drug release, it was almost been done till more than 40 hours, up to close 50 hours. And it has clearly shown that there was a constant abrupt increase up to the 18 hours. And after that, it was significantly showing a constant release up to the 50 hours. So that shows that when patient is going to have this through the inhalers or particular nebulizer system, then it can have a sustained release or a prolonged release for a longer duration, and the patient need not have to worry about taking this medication over and over. And that has clearly shown it has a high entrapment efficiency. It has also shown high affinity towards the hydrophobic domain of the nanoparticles. And finally, it has shown the sustained release. So what we have done now, we have used two different cell lines. For the oxidative stress, we have used the BESB2B cells, which is the epithelial cell line. And we have clearly shown that the different concentration of the rutin when we have used starting from 1 micromolar to 20 micromolar, it actually have reduces the percentage viability of the cells. And when we have looked at the nitrate level, which is an important contributor to the oxidative stress, on the treatment with the rutin, it has significantly been reducing the level of the nitrates, starting from the concentration from 1 micromolar to the 20 micromolar. Then we moved on further looking at the various cellular reactive oxygen species in the LPS-induced BESB2 cells, because LPS is the one which brings out a significant inflammation, and we use the DCFDA method. What we can clearly see here is, so you can see that in the control, there is not high fluorescent intensity, while as the treatment goes up from the 1 microgram per ml of the rutin liquid crystalline nanoparticles to up to 5, you can see that intensity is actually increasing. And that was clearly showing that how actually the reactive oxygen species are actually being contributing into this process. So what we have done next is we looked into further into the apoptosis, because it was really important whether the cells are being killed by these rutin liquid crystalline nanoparticles or not. So we have clearly seen that when you see that moving from the control towards the highest concentration of the rutin up to 4 micromolar, the intensity is increasing. And that clearly shows that the cells are not actually being, the apoptosis is not really happening. So cell death is not really happening here. It means the rutin is quite safe. And then when we have looked at the anti-proliferative effects of the rutin in A549, we have also seen that with the higher concentrations of the rutin from 2.5 to 20, it reduces the percentage viability, as well as it reduces the number of the cancerous cells. This was just a basic MTT assay which everyone do to start their study. And that was clearly showing that the cancerous cells were really having a high impact. And then when we looked into the anti-migratory effect of the rutin, that was clear using a wound scratch assay on the cells. It was clearly showing that the percentage wound closures was happening as higher as possible as the concentration progressively increased from the 2.5 to 20 micromolar. And the same thing we have even observed with the migration assay as well. When we looked into the MMP9 enzyme, now MMP9 is a metalloprotease protein. And this is one of the significant contributor to the mechanism in this disease. And to do that, what it happened is when we looked into this, we have done a Western blot and we treated with the different concentrations of the rutin. So when you can see that as the concentration is increasing of the rutin, you can see that MMP9 levels are actually reducing. So that also has shown and provided us an insight that whatever is happening with the oxidative stress as a one axis, while on the top of the thing, if we look on the migration and proliferation on the other axis, MMP9 was one of the contributing factor. So when we looked on the cells, they were also been clearly showing the morphological changes. And you can see that this is the control, and they are quite healthy cells, and they all are cancerous cells. So you can see that morphology, the shape is really needle shape, the spikes coming out, they're growing very well. While as we are treating with the different concentration across the line, you can see the cells start getting dead. It means the apoptosis was happening and the cancerous cells was actually being dead. So apoptosis was also being checked through the fluorescent microscopy. And you can see that in the control, there is not as intense fluorescence. So this is annexin 5 FITC, and this is DAPI, and this is the overlay of both. And you can see that clearly that the cells, the apoptosis were not really happening with the higher concentrations of the rutin as compared to what's happening in the control, where the cells are being having a significant survival. So in the nutshell, there were two different conclusions we have concluded from this. The most important thing, this was a different study which is already been published in the toxicology in vitro. We have seen that with the lipopolysaccharide induced inflammation, probably we are hoping, which we are testing at the moment, is they are being targeting the toll-like receptors. Now, when they target the toll-like receptors, they actually probably be hoping, because we already have looked into INOS, because nitric oxide we have measured. So that clearly shows, yes, it gets upregulated, while with the treatment of the rutin, it reversed the cycle. Not only just the nitric oxide, when we give the liquid crystalline inhibitors, it reverses the cycle and it stops the cell apoptosis. On the other hand, when we have looked that when the A549 cell line, so the previous study is done in the BASB2, which is the epithelial cell line, while this study is done in the A549. But we have to understand, when we talk about the respiratory system, the epithelial cell is the major line of the defense. And these epithelial integrity is really important. If these epithelial cells started undergoing hyperplasia or any kind of the things, then the cell leakage or any kind of the mechanistics hazards can happen, like the leakage or the oxidative stress or a DNA damage. So likewise, in A549 cell, what was happening is there was because of the oxidative stress or because of some other reasons in the cancer cells, there's a significant DNA damage. And that DNA damage is being controlled by the NF-kappa B, which is the very significant mediator in most of the chemical reactions. And that was leading through the apoptosis. So currently we are looking into the insight into the in-depth mechanism with PR3 kinase and all these things. And that is leading to the cell proliferation through MMP9, which our study have shown. And then we have clearly seen that when we give these inhibitors actually inhibit and stop the apoptosis. We have clearly seen in both the cell culture experiment as well as with the fluorescent imaging, the microscopic images. And these apoptosis, when the cell death stop, it automatically actually with the reduction in the MMP9 level, and it increases the extracellular matrix degradation, and that leads to the migration and prevent the disease pathology and the disease features. So this is just a small part of the study which my lab currently is doing, but we are also looking in different drugs at the moment, such as berberine. We are also a paper is about to come within another drug called lensclunaris, and rutin, nerogenin, and cilastrol is the another one. So this was the paper which came out in terms of the oxidative stress and apoptosis in the bronchial epithelial cells in vitro. And this was a study, actually the previous, this study was actually a collaborative study between the International Medical University Malaysia with the NIH. And so the students actually have prepared the formulations back in Malaysia, and it's been sent to my lab where it was tested on these two different cell lines. And likewise, there are various formulations we have worked together. So at the end, I would like to acknowledge particularly the Graduate School of Health where I'm based at, and the people I work with, particularly Mary and Kylie, with the Center of Inflammation where I'm the node leader for the drug delivery node. So Phil Hansbrough is my mentor as well as the head of the Center for Inflammation I work with. Keshav is the one who helped quite significantly with number of the cell culture experiments which we do. Gang actually look into the remodeling studies, and we are planning to look into the collagen and the fibronectin and fibrin now with these formulations. And these are my PhD students and the master students. I would also like to acknowledge quite a significant number of people I collaborate with, and they play really a very crucial role here, because I don't think without them it would be really able to attain what I have achieved so far. So they even start from UK to Australia to Brazil. And I believe Professor Teresina and Antonio, they might be live with us. So my best wishes to them actually, because they've been a great support as well in various experiments and stuff. Dinesh is really miraculous because most of the formulations come from his lab, which we test in my lab on the various cell lines. And also Anand, I have just started working last from 1.5 years. And Pradeep, Professor Vinesh Pillai is, he just passed away last year, while Pradeep still helps significantly with the molecular modeling studies in South Africa. Anand, I've just started working with him, and we recently had a paper about to come on in the ACS Omega. So exactly on the same lines, we have worked on the different nanoparticles, and all are from Jordan. On top of that, Hari is also been presented yesterday, I believe. So he's the one who is a medicinal plant expert, and he's the one who suggests quite a number of phytoconstituents to us. From the Korea, I have Renu and Hari I work with, and India, I work enormously with quite a significant number of institutions from the Manipal to NIPER to MDU, LPU, and Shulini University, and Savita, where in few of the university, I'm on a conjoint position. And the last but not the least, I would really like to thank Ronan from Aerogen, and he's also a part of the Royal College of Surgeons, Ireland, and the School of Pharmacy and Pharmaceutical Sciences. So he is now helping testing our formulations through the nebulizers and optimizing the parameters. So once it would be done, we would be able to take this into the translational stage, because we're already doing the mice models, but we would like to see how exactly it is affecting in the humans, because no point doing the research, making these nanoparticles, doing the physicochemical characterization in vitro, and just publishing the paper while it's not going to the next level. So Ronan is really a great support, and he's helping translating into that. So we're going to send some formulations to him, and he has supplied some equipment, which gives actually quite significant parameters, which we can test through the these formulations. So I think that's all have I in my presentation, but I would also like to have my sincere thanks to Dr. Rajiv Daya and to organizers of the this conference of the Association of Pharmacy Professionals. So it was really a pleasure actually speaking and sharing my research with everyone. And look, I'm really open. If anyone had any questions, I'm more than happy to take now. But I'm really sorry because I was being offline most of the time. I was a bit flustered. It's just a holidays period been starting here. It's just the Christmas on the peak. People are coming in and out. So it was just a busy time. So just excuse me for that. But I'm happy to take any questions if you got. And thanks again.
Thank you. Thank you, Dr. Kamal Dua.
Professor Jaybalan, Dr. Dua session can be completed now only. So he will not be available.
Okay, okay. So is there any questions from panelists and chairpersons to be asked to Dr. Kamal Dua because of or otherwise we'll move on to the next session, I think.
Yeah, Doctor, I want to ask one question, Jaybalan sir. May I ask?
Please proceed.
Yeah,
Dr.
please. Yeah. Dr. Kamal Dua, sir, you explained your research work in very simple language, and I appreciate the effort, the research work you did for humanity. My first question is your work is on respiratory disease. Your complete work, research work is on respiratory disease. How can it helps in treatment of COVID? Because now everyone is, yeah.
It's a very good question, Professor Goel. And I think this question has been asked to me in every presentation I've been delivering from last 3 months off and on. So before actually I started working on the targeting phytoconstituents, my lab was focused in understanding why the patients with asthma and COPD are more susceptible to influenza infection. In simple language, when a patient is having asthma or a patient is having COPD, which is emphysema, and they get flu on top of that, their condition really exacerbates. And it's really hard to get managed. And this is what we are seeing in COVID as well. So if a patient is, I'm not talking about a healthy person getting a COVID. I'm talking about a person who is already having asthma or COPD and getting COVID on top of that, their condition exacerbate. While if we see as a control, a normal person getting COVID, they normally recover easily. While their oxygen level also goes down, their pathophysiology also change, and the normal process of any infection is same. Inflammation happen, there is a burst of the inflammatory cells, particularly macrophages, and particularly it depends on the condition whether you have asthma and COPD. In asthma, you get a burst of eosinophils. In COPD, you get a burst of the neutrophils. So yes, so what we have identified is there are two different microRNAs. One is the microRNA 21, and other is the microRNA 125A and B. These two microRNAs, they really get upregulated when anyone gets infection plus disease. In control, they really don't have these levels up. So when we give inhibitors of these microRNAs, they reduce the level of these microRNAs. And when they reduce the level of these microRNAs, it automatically reduces the level of inflammatory cytokines. I don't have time. I could have shown that slides to you. I can show if I can find quickly, if you give me a minute. And because of that, it automatically actually reduces the disease pathology as well. So you can imagine like how, if you target these microRNAs, it is so beneficial. And we have submitted a grant proposal in exactly in the same way, because I was having already a proof showing in flu infection that if we target these microRNAs, it reduces the pathophysiology. So in COVID also, if we manage to target those microRNAs directly rather than being digging from the scratch and thinking this is the thing gonna help or not, then that thing might can help in identifying the potential therapy. But to be honest with you, everything takes a lot of time here. Yes. It's not easy because first of all, you need to test these things on the mice model, and the mice model itself, it takes a lot of time. And after that, when it comes to the human clinical trials, it is another problem. Okay, let me show you these. Let me share this with you. So, okay, can you see this slide? So yeah. This is just in a COPD with infection. So, and these are the human cells which we got from the COPD patients by bronchoscopy. Okay, so this was done by a clinician and they gave the cells to us and we treated. So look here. So if we look here, so the three different groups, healthy, COPD, and smoker. Healthy is the one who don't have COPD. COPD is the one who only have disease. While smoker is the one who don't have COPD, but they smoke. Because COPD is a disease which caused due to smoking. And you can clearly see that all the inflammatory markers, IL-6, CXCL8, TNF-alpha, they are up in COPD. See how high their levels are. And because we're talking about infection, so if we talk about interferons, they are suppressed. So what we have done here, I'm just quickly showing you. So we gave these inhibitors of microRNA 125A and B. And I'll show you straight away this. So look, when we gave the microRNA 125A and B inhibitors, it started reducing the levels of the inflammatory cytokines. And if I go just on the concluding slide, this will give a whole picture. So when a patient having a emphysema or asthma, I will show you in a minute asthma as well, and they get COPD with flu, they get high levels of microRNAs, and they reduces A20 level and mitochondrial antivirals. And that actually significantly increase NF-kappa B. And because of that, the inflammatory cytokines increase. And when we inhibit these, it reverses the cycle. And when it reverses the cycle, it completely stops the thing. So imagine now, instead of flu, you have COVID. So this cycle may work. I'm not saying I'm a scientist and I'm a really an early career researcher, and these are just the thoughts. And this grant is still under review. Finger crossed, it has not yet rejected, and I hope it's not. So there are avenues. You can explore the same things into the different diseases. Doesn't matter what kind of the infection is, whether it is flu, whether it is COVID. The main problem with any kind of infection is it triggers the inflammatory response. And
One more question, Dr. Kamal Dua sir. You said routine has anti-inflammatory action, and it also inhibits platelet aggregation.
It does, but it won't work on platelet aggregation. This was just been identified through the literature. So we only have worked on the respiratory context, so not on the platelet. I don't have any idea, but it was just a literature.
Okay, it is given in literature. But yes, it is a herbal extract. It is a herbal extract routine. Right. So is it possible to use this routine for the treatment of COVID?
Indeed, very much so. The only problem is when we have tried to do these things, you need a specialized laboratory called PC3 facility. And to get the approvals itself, it takes 6 months here in biosafety. So we have put the proposal, and if that grant being successful, then if they allow us, then we might be able to try any kind of nanoparticles. Doesn't matter it's routine. Any nanoparticles which trigger the inflammation, every disease, the basic thing which I will tell everyone, doesn't matter what kind of the disease you have, whether it's a brain, eye, lung, kidney, every disease has inflammation. And the underlying mechanistic problems are the inflammatory cytokines, or whatever you say, mediators, they are same. So if you manage to target those with anything, you don't know the routine. What I'm showing in my lab is respiratory because I work on that. And if I give that to you, your lab is working on kidney disease. If it has shown that it inhibits the inflammation, it might inhibit the inflammation in your disease you work as well. So the answer is yes and no. I have no idea until it's been tested.
Okay, thank you.
No, thank you, Professor Goyal.
Thank you. Thank you.
Thank
you
you.
explained all my queries very well.
Thank you.
Thank you, Dr. Kamal Dua. Thank you, Dr. Anju Goyal. And we move on to the next speaker, long-awaited Dr. Vechiselvan Subramanian. Sir, please, if you start your presentation, then we can move on to the next presentation, sir. Yeah, you please start your presentation, sir. No, sir. You are? Hello. Sir, you're on mute, but your audio is not reaching us.
Sorry, you asking me something?
No, sir. The questions are complete. And there is one general question in the panel chat box that I read out. If you can answer, then it's not a problem. Otherwise, we will put it to the other speakers. One, Dr. Vijayalakshmi from Anna University, Trichy has asked that, sir, is there any chemotherapeutic agent is now available to cure stage 4 lung cancer? Since it is related to lung, I am directing this question to you. This is from Dr. M. Vijayalakshmi. She is an assistant professor of BITS campus.
Right. It's really an interesting question. So yes, and so the most of the common treatments which are used in the stage 4 non-small cell lung cancer include the cisplatin, or I believe it is called carboplantin or docetaxel. People also use carboplantin and paclitaxel, and these are the kind of the drugs which have been commonly used, while my lab particularly focus all on the herbal medical herbal constituents or the phytoconstituents into the liquid crystalline nanoparticles. And it's really difficult because the stage 4 is really a piece of the research, is a piece of a cancer where it is reached to the peak. So only the chemotherapy which can help is this cisplatin or carboplantin, what you call, or docetaxel. These are the drugs which are commonly used. But what we get the cell lines are the not really up to the stage 4. So it's really difficult to trigger those, while people are nowadays actually trying extracellular vesicles. And these extracellular vesicles are really a good form, and they can target any kind of the degree of cancers than these chemotherapeutic agents. So I think that's all I can add in terms of your question.
Thank you. Thank you, Dr. Kamal Dua. And you may take your leave, please, as you are having some problems. And we now move upon the next speaker. Are you able to connect with me, Dr. Vettiselvan, sir? Otherwise, we'll move on to the next speakers. Your audio is not receiving to us. We are not hearing you. So this has become a senior junior struggle. What we felt in Annamalai University, I think is time has come back. So we will wait for your lecture, and I can move on to the next speaker, Dr. Rohit Kumar Verma. May I please request my organizing team colleague to recite the profile of Dr. Rohit Kumar Verma. And Dr. Menon is also waiting for a long time. I'm sorry for making you to wait. And now the time comes for Dr. Rohit Kumar Verma. Please recite the profile and call upon the presentation.
Thank you, sir.
Yeah, hold your mic.
Yes, sir. I would like to share a profile, Dr. Rohit Kumar Verma. Presently, he is working as a head of Department of Pharmacy Practice, School of Pharmacy, International Medical University, Malaysia. He did his doctorate from National University of Malaysia. His area of interest is ocular kinetics, drug utilization evaluation, obesity, clinical trials methodology design, and community pharmacist-led interventions. He has published various research review articles in reputed journals. He has received a Best Poster Award from E-Learning Festival IMU, and also received Good Clinical Practice Qualification Certificate from Ministry of Health, Malaysia. He has attended various national and international conferences. I welcome you, sir, and share your research experience on professionalism and ethics. Now over to you, sir.
Yeah, my request to Dr. Rohit Kumar Verma is please make your presentation a little faster so that we can cover up the remaining scheduled events also. Please. Yeah.
Yes, surely I will take very less time. In fact, thank you very much to all of you for inviting me in this convention. I feel really, meaning I was thinking to be like a number of students will be in this session. However, I feel that seems like very few number of students are here. almost most of the professors and doctors are here in the queue. So I am really not sure how much my presentation will be in your favor, because I have designed these all slides in favor of my students of B Pharm and M Pharm graduates. Generally, to be take care of them should not involve into any unprofessionalism and unethical behavior. So definitely in this way, I would like to proceed very fast and we'll finish on time. So the question arises here, has undergraduate education lost its way? So why we are asking this? Because we have seen that currently the number of other universities and good universities actually have really stressed out because of demand of stakeholder satisfaction. Each university is actually is under pressure to achieve a rank, to achieve a good what is called of milestones in their areas. So somehow from the management to the staff to the students, everyone getting some experience to get a lot of pressure to achieve targets. Okay, and when we are trying to and strive to achieve a target, definitely sometime we go through some different path to achieve the jobs in easy manner and unprofessional manner or unethical manner to grasp the same number, and here the things go wrong. So the importance of this lecture was whoever is strive for the best must be adhere with the professionalism and ethical practices. The reason behind of stimulation is this: if one's unethical behavior is spotlight by the agencies or by the government or by the system, actually that career lost everything in his life. Okay, so this is the importance of this lecture. Sometimes we ignore it, because in fact if you see the university grades, the university report card do not have the moral grades. There is no moral grades marks of A, B, C or what you have. Part of the reason we have a impression that the most of the student already have achieved all morals into their school level, and once they come to the university they should learn what is called professionalism and involve into the professional characteristics and behavior to uplift them, rather than we are not here really to correct their morals in general. So almost in every university this morals, behavior and ethical and professionalism are most ignored part most of the time, and we are always focusing on, oh yes, how many papers you have published, oh yes, how many grants you have got, oh yes, how much perks you have got in a year. But we have lost our value. We have lost the sense how we should supposed to be useful for community, how we should be at the good level in scientific community to get an actual reputation of our own face. So I give you the example here. He's the Dr. Harold Shipman, is the doctor in UK. When he was the student of MBBS, he was caught while selling the drugs into his peers. And when his case went to the dean level and principal level, they both have suggested that this student particularly is having a very good credentials, so please excuse him, otherwise his career will be waste. And somehow he was able to manage, complete their graduation, and later on, once came to the practice, he was responsible for 250 deaths knowingly, because he used to give the excess painkillers, the excess dose of painkillers to the patient, those who are bedridden patient, meaning the patient those who are for longer period of time ill. He loved to give an excess dose of the the painkillers and to kill them. So in the one speech he stated that I love to clean the beds. So when he stated the statement, I love to clean my beds, I hate the patient who was bedridden, the police got attention, the security agencies got attention on his statement, and they got doubt on his statement. They started following up to 6 months, and they found that this fellow is involved into 215 casualties. Okay, and which has given a very much, even including royal societies of medicine and also societies, those who are developing the UK graduates as a medical students, is left the question mark on them. How come this particular person has completed 5 years of graduation and yet not have a simple morals in his mind? How the university has graduated him, on what basis, and why the university not able to catch him on the time, so we can save the at least 200 people. Okay, so somehow we consider you got the first rank in the class, so you are the best student. what the way our teachers have sometime perception, including our management have such perception that a student who is scoring highest marks in general is the most good person. But fact is, it's not directly, the grades are not directly proportional to your professionalism and ethical behavior and being a good human being. Both are different things. A clever man can get a higher grades, but maybe is not a good human beings. Okay, so we have to understand here is the fine thread behind. So what is profession? When the profession comes, we used to say be professional, behave like professional. But when you ask your student what is profession, actually they might not able come back to you. So illustrate them, the profession is somehow is the two part of what is knowledge, and another part is the behavior and commitment to the service. Like a teacher have a knowledge and commitment to teach to the student. If both is combined, you are the best academician. Okay, so similarly, University of Education, you see the most of the universities now pop up they say that I'm the most private leading university, I'm the first rank university into the area. And they pop up some good faces of the academic people, those who are most handsome and beautiful. And similarly, the student group of like boys and girls, they pop up here like this is the most good university for the study. In fact, they are bound to showcase. In fact, they are bound to showcase to run their business. Okay, but we as a student, we have to understand what university, when we entered into the university, what is giving the sense to us to develop us as a behavior, characters, values, traits in ourselves, rather than as a good company of two students or four students. This is not university. You are spending the huge time. You go to the each corner of the university, you utilize each service in the lab or library, or you met with your teacher, you share your thoughts, and you share your experiences with them, and ask them how you have go through the long journey. Like you have done your PhD, I would like to proceed the PhD. How your PhD has given the experiences to you, to inspire yourself, rather than self-limiting yourself with the 4 or 5 students. So professionalism generally have a characteristics of a good communication, a good teamwork, critical thinking, following the ethics, and last but not least is the humanness. So the question arises, can unprofessional conduct during the student days lead to trouble while he is practicing pharmacist or physician in later life? The answer is yes. We have investigated the candidates, those who caught into the plagiarism, those who caught into the wrong publication, those who caught into the some historical manner of of involved into the narco drug supply or any crime. When we have investigated their school time, the school teachers say yes, when he was in the year 3 or year 4 or year 5, we have penalized him because he has done this activity during he was 7 years old. Character, attitude, and behavior is not easy. It's not really easy. And I can assume that a lot of university professors and teachers are really struggling with the one factor associated with the wrong students is bad attitude. And this bad attitude is tough to correct in the overnight process, because teachers have a lot of pressure to teach also, rather than correcting the behavior at this level. So looking here, we have to be take care of, once we go to the system, like once our students life over, we are the part of the system, we are the academician, we are the researcher, we are the businessman or anywhere. How the people look on you is first is altruism. Altruism is your caring behavior, how you look the others, how you are helping to others, how much you are doing good for others. If you have a BMW, Audi, or good funds in your pocket, no one is going to get impressed from you. I tell you, the reality is that everything belongs to you, not to the third party. If you have, it's up to you. You are not going to donate the millions of dollars to the normal public, right? But if you are useful, if you are valued, if you are making others' life valued, they will give the respect to you, no matter you have 0 cent in your coin, 0 cent in your pocket. So the question is, how to be become valued? How the public give the value? How the you see the businessmen like of Ratan Tata and other businessmen. So when the good businessman face comes in your mind, how the Ratan Tata face comes first? Because somewhere is the valued rather than business. Okay, so public is very intelligent to select you and very intelligent to identify your traits and characteristics. here is of like integrity and other things like we must be honorful for others and we should have a great respect and admiration of others. Also, we should be admit our own errors. It is very tough to admit, but we should accept our own errors whenever we are wrong, and we should deal with confidential information discreetly. I will tell you later where and how it is important. We must be caring and compassionate. We must be kind and helpful, showing to others how we are treating to the patient and other individuals, everyone notifying you. Actually, the many time your actions was observed rather than your words. So when you are observed any action of the any individual, you can easily identify his traits rather than going on his words. Okay, you see how he act, how he behave, how he talk to others rather than to you. You can easily identify the real face. So respect. When you are in the peer, are you respecting to the peers or not? Or only you are respecting to your manager or line manager or the boss or the principal or deans, or really you are respecting similar, giving respect who is getting the same salary and having the same designation? Are you respecting the same as you're respecting to your line manager? The answer is no. Okay, so here is the fine thread. We should understand, right? The respect should not go according to the designation. Respect should go according to the value of the person who have. So again, does not disturb the small group sessions. Yes, this is also important in a student learning time. I have mentioned like we should not misbehave while in the small group learning. So give respect and get respect is a simple phenomena. Taking responsibilities of the most of the time when the line managers usually may say to you, you did wrong this thing. You are responsible for this bad event. In fact, when the command itself is wrong. So the question arises here. It is very easy as a line manager to me. I am also head of department. I am controlling 24 individuals right now. I know where I should admit my mistake itself. I should be responsible as a head. I used to say this is my mistake here. Okay. So similarly, a very few line managers may have guts to accept that I'm here wrong. I'm wrong here. So we should be role model for the young people. And like if we are in the office, we must be arrive on the time rather than like, directors, deans or any organization people, they are coming 10 a.m. but they are asking that who came at 30 a.m. and late. So this is really not a good role model. Say we must be on time itself first before to say someone that you are late. So accountability for deadlines, complete assignments and all. Like if I never send my work like you have to do at a 9 a.m. or 3 p.m. I never send any email like that you have to finish according to the AM. Okay, unless until it is not urgent. So many times the line managers are keeping their work up to the 30 days with them. When suddenly the higher management asking them, where is this record? They give the very tiny time to the downside. You have only 24 hours time to complete this task. But this is not a fault of your lower employee. This is fault of your who has not cascaded the job on time. Okay, so this is accountability. You should accept the accountability of any falsification, any error into the documentation or any problem into the documentation. We as a head are responsible for that. So again, the excellence. How you consider you are the excellent here, meaning we have to set our goals. We have to be a like of self-critical into the thinking and identifying our own areas where has to fly to achieve the goals. We should be a good leadership, having a good leadership, and we should be, the leadership is only possible when it is you are self role model for them, for the next party. Very fine thread here. The people get confused between the empathy and sympathy. We as a healthcare professional, generally in a ward, when the person get die or any casualty happen, you can see that many relatives have started crying into the ward. But being a doctor, being a pharmacist, we are not allowed to wet our eyes. Okay, in fact, we are keep on continuing the job what is assigned to us. So wet eyes is really, somehow emotional attachment. And why we are not allowed to cry into the ward, part of the reason is we are healthcare provider. If we ourselves will started crying in front of the patient, the patient actually, who is the another patient who is looking on you graciously, that you are the person like God for him, and he is looking a hope from you that you will provide the life to him. But if you are also emotional and also started crying and your eyes are also wet, the next person who is closely observing you, that is patient, may get disappointed while looking on your face. So always be firm on your, show the empathy, you no need to show the sympathy there. Humility is another good component, like of the quality of not thinking that you are better than other people. You always make yourself better, but you not necessary to tell that you are better than A, B, C, D, E, F, G, H. And out of 14 candidate, where is your rank? However, in human beings, we always discriminate the people. You are the champion, you are the first, second, third, fourth, five, just in the, because we love that discrimination. We love that discrimination. Almost being a parents, when you have never giving ranking to your own kids, right? If you have the 3 kids, they're all 3 kids coming back towards you, mama and papa, who is the best? And you always convince them you all are best. You know worries, you are the best in this, you are the best in this, but you never give the rank to them. But we as a teacher, we as the in business, also in a corporate, we always love the rank, meaning you are the number 1 employee, you are the number 2 employee, you got 3 points less, you got 1 point more, something like, to hurt them, not to create good values in them, rather than just discriminating them with ranks. And that ranks is actually never useful for the further system. So what is ethics actually? The question comes, what is ethics? Ethics is of as a civil code, a good behavior of like how the one individual is think, act, and behave. Okay, and the further explanation here is the code of ethics. After graduation, you are getting one code of professional book, and that professional books are usually having a code of conduct behind. And many people, even in pharmacy, those who graduated, they never look their green book code of conduct, where the number of code of conduct statements are mentioned that you will adhere with the practice, right practice and good practice. So that practices and code of conducts are very important for you to follow during the practice. So what are the ethical values being a professional? We must be able to keep the patient confidentiality. Okay, always should be truthful for the patient. Okay, do not harm any individual knowingly, and we should do our best for the patient and disclose whatever is really essential to disclose, otherwise no. Implementation, yes, you are the faculty here, most of them, so you can see like professionalism and ethics in generally in our teaching. We are doing some seminars like in small teaching, we sometimes we have some small teaching, so we are giving some sense to them, whatever the professionalism involvement is here, whatever the ethical involvement is here. So our students somehow they are learning, but we believe that this learning is from childhood, and always the student must be brushed up from the school days rather than at the university level. So some professionalism and ethical issues are discipline, plagiarism, cheating, hacking of computers, and fitness to practice. I tell you one here, like discipline. We love to say you should be disciplined as a student. So, or even the faculty should be in a dress code. Our attendance should be good. Disruption of class is really not acceptable. We love to showcase them that you are not allowed into the jeans. Okay, you are not allowed into these hairstyles. You are not allowed with the tattoos and piercing. Okay, you are not allowed with the earrings and all. Okay, you are not allowed with the jewelries and excess, and you are not allowed into the campus with sleepers like this and t-shirts like this. The question arises, sometimes students get burnout. When students get burnout and they say, we really don't like the campus or university who judge us on the basis of attire. And many times I am the student advisor as well, and sometimes students come, they say like, the time has been changed, sir. You guys has to look that attire has do not meaning any with the behavior and character. And it is not necessary if I am wearing the jeans, I'm having a bad character and trait, something like that. They defend like this. So I love to ask the question to them, when the campus interview comes, when the placement comes, why you in the white shirt and black pant and black tie and coming in a very nice manner with the essence and perfume? Why you are not coming on that day with the t-shirt and slipper? They are always be silent. So we have to tell them, we are only requesting them to be in the dress code in the campus. You are free and you are allowed to wear anything in your own personal or private area where you are living. We are nowhere is interfering you. Okay. So look on the army, look on the navy, look on the air force, look on the hospital premises, those who are having white coat. When the person, the public close their eyes, how they look about you, how they perceive when they close their eyes, how they think about the doctor. The doctor looks of white coat on very disciplined candidate, very enthusiastic to for the helpful for the patient and all like that. They are not willing to see the like a casual person and here and there person. So we have to tell them the importance of dress code in our profession. Actually, disruption of class is another challenge. So we have to understand this is really not a good professional behavior, like talking in a class, making noise in a class, using mobile phones, sleeping in a class is really rude. We used to tell every time, and every time some teachers used to say, but unfortunately the students are understanding in a different manner. Plagiarism is very high on the peak. You can find that number of people have published the article has been retracted later. Okay, sometimes they put themselves into the big trouble, and yes, with the one email itself the career can be vanished. Okay, so they should the harmful events associated with the like of cheating, plagiarism and other issues at the student life. Some guys now you know like they are very good and they feel that they are very good in computers, so they would like to hack the computers of the teachers, or like if teacher left his laptop and there is no password is already there, they would like to swim into the emails to get a question paper. Such kind of attempts actually can be harmful and under code of conduct. So we anticipate the high standard of professionalism conduct is expected of you as a student at all times, both on and off campus. So that is all from my side. Thank you so much. Sorry, I was a bit fast just because I was requested to go fast. Okay.
Yeah, thank you, sir. Thank you.
Yeah.
Thank you very much. We have 2 more lectures to be completed within time, no? So that is why we were hurrying you. Please stop your sharing, and then we'll move on to the next speaker. We'll move to Dr. Vinita.
Sir, can you hear me?
Yes, sir, I'm hearing you.
Okay, sir. Just you can go ahead with other lectures. Maybe I will take the last session. No problem. I will wait.
Yeah, please, sir. Sir, this has given a long many time to stay with us. So please stay, and she is also waiting right from the beginning. And
it's okay.
I'll
I will take the last session. No issues.
Yes, sir. Dr. Vinita Bharathan Menon. And I request my organizing team colleague to recite the profile of Dr. Menon, and please ask her to start her presentation.
Thank you, sir, for the opportunity. It's Dr. M. Ramesh. I'm professor and head, Department of Pharmacy Practice from JSSHR Mysuru. A very good afternoon to you all. The next section is on role of pharmacist during COVID-19. And the speaker is Dr. Vinita Bharathan Menon. I take the privilege to introduce the speaker. To introduce Dr. Vinita, Vinita Menon obtained her bachelor's degree in pharmacy in 2009 from Amrita Institute of Medical Sciences and Research Center, India. Later, she obtained her Doctor of Pharmacy in the year 2014 and Doctor of Philosophy in the year 2020 from JSS Academy of Higher Education and Research, Mysuru. In February 2018, Vinita joined as a lecturer in the Faculty of Pharmacy and Pharmaceutical Sciences at the School of Pharmacy, Monash University, Malaysia. Previously, during her tenure as a doctoral fellow for 2 years, She was involved in bedside teaching for postgraduate and undergraduate level pharmacy students at JSS University. Apart from teaching, she was also actively involved in research, mainly in patient medication and drug safety and health outcomes research. Also, she has worked for 2 years as a clinical research coordinator in phase 3 sponsored clinical trials. And her clinical experience includes serving as a clinical pharmacist, working closely with physicians and other healthcare professionals, and guiding pharmacy students during their clinical placements. Besides this, she was also previously employed as a registered pharmacist at a multi-specialty hospital in India. Overall, Vinita has more than 5 years of experience in academics, clinical practice, and research with a strong background in applied therapeutics, clinical pharmacy, and drug safety. With this brief introduction, I welcome Dr. Vinita and invite her to deliver a lecture on the topic, role of pharmacist during COVID-19. Over to you, Dr. Vinita.
Thank you, Ramesh sir. Before I start with the presentation, I would like to thank all the members of the committee of APP as well as the president for giving me this opportunity. Is my screen visible to you guys? Is my slides?
Yeah, we are able to see.
Okay, thank you. So as Ramesh sir highlighted, I would be going to present about the role of pharmacist during COVID-19, especially from a clinical perspective. My name is Vinita and I'm a lecturer at Monash University in Malaysia. So moving on to the lesson plan, as a part of today's presentation, we would be looking at a brief overview of COVID-19, as well as its timeline in Malaysia. We would look at the mode of transmission and the clinical presentation, the risk factors for high mortality rate among the patients, the treatment options for COVID-19, challenges faced by clinical pharmacists, as well as the role of clinical pharmacists amidst the pandemic. So as you all know that in December of 2019, we came across a respiratory illness due to a novel coronavirus, which was first identified in the Wuhan province of China. Right now, this respiratory illness, which is known as COVID-19, is a global pandemic. And as of now, we have 75 million cases reported worldwide, with 1.67 million patients dead across 216 countries. With regard to the timeline in Malaysia, we had our first patient identified as COVID positive on 23rd of January. On the 27th of February, we had the first wave of COVID-19 in Malaysia with 24 confirmed cases, 2 active and 2 new cases. On the 18th of March, the government over here declared the movement control order. We had 728 active cases at the time with 117 new cases. On 7th of April, we had the highest peak of COVID-19 cases in Malaysia with 2,579 active cases and 110 new cases. As of now, with today's statistics, we have a total of 90,816 cases reported in Malaysia, with around 75,000 patients recovered and 432 patients dead. With regard to the transmission and the clinical presentation, we know that like any coronavirus, COVID-19 spreads from one patient to other through coughing and sneezing, through close contact with the infected person, and through touching any contaminated surface. You must be knowing that the incubation period for the virus ranges from 11 to 14 days, with a median time of 4 to 7 days from the date of exposure to the onset of the symptoms. Now, as seen in the slide, some of the common presentation of COVID-19 includes cough, shortness of breath, and fever. Now, the disease may present as asymptomatic in some patients, whereas in some it might be mild disease, whereas others might present with moderate to severe disease, including pneumonia, respiratory failure, and death. The mild disease as I mentioned previously, is even mild to moderate disease actually, as I mentioned, is the most common, and it is actually seen in 81% of the patients. And usually these patients may present with upper respiratory tract infection as well as non-severe forms of pneumonia. The severe disease, which is characterized by dyspnea and hypoxia, is usually observed in 14% of the patients. And the critical disease, which is characterized by respiratory failure, shock, and multi-organ dysfunction, is actually rare, and it's only seen in 5% of the patients worldwide. We have to keep in mind that the fatality rates are highest among patients with advanced disease, especially those with underlying comorbid conditions and elderly, advanced age. Now, moving on to the risk factors, according to the CDC, the risk factors for severe illness from COVID-19 includes age. As I mentioned, anyone above the age of 65 years have a poor prognosis following infection with COVID-19. Other risk factors include cardiovascular diseases. As shown in the slide, patients with cardiovascular disease have 5.8% higher mortality from COVID-19 in comparison to patients without cardiovascular diseases. I have highlighted some of the statistics with respect to some of the more common cardiovascular diseases. You can see that any patient who suffers from coronary artery disease or heart failure or cardiac arrhythmia definitely have a higher percentage of mortality in comparison to those without the disease. Another risk factor includes cerebrovascular diseases, particularly prior history of stroke might increase the severity of illness from COVID-19. Diabetes is another risk factor. We have a trial which was conducted, particularly the Coronado study, which highlighted that patients with diabetes, 1 out of 10 died within a week of hospital admission, and 1 out of 5 with diabetes were intubated and required mechanical ventilation. The study found that poor blood control is not the risk factor for higher mortality, but rather it is the diabetic complication and the age of the patient, which is predisposing the patient to poor prognosis. As one of the other speakers mentioned, respiratory diseases like asthma and chronic obstructive pulmonary diseases can increase the risk of severity from COVID-19. We have found that patients who have moderate to severe asthma are at higher risk of developing severe disease from COVID-19, and patients who have chronic obstructive pulmonary disease are 2 times more likely to develop the severe disease. This could be due to the underlying inflammation that is associated with these respiratory conditions. Another risk factor is cancer. Any patient who have current cancer are at high risk of developing severe illness from COVID-19. However, at this time, we do not know whether the past history of cancer can increase the risk of mortality. Similarly, chronic kidney disease, any patient who is on dialysis is found to have a weaker immune system, making it harder to fight infections. At the same time, patients who have received transplantation, they almost receive immunosuppressants, which makes it harder to fight infection. I highlighted these risk factors, particularly for clinical pharmacists, to pay more attention to these patients because they are at higher risk of mortality rate from COVID-19. And as clinical pharmacists, we have to pay specific attention to these special population. Now, with regard to the treatment, the main objective of COVID-19 treatment is to relieve the symptoms. So as far as we know, the mainstay treatment involves use of pain relievers. The most commonly used pain reliever is acetaminophen. In outpatient department, we tend to use a lot of NSAIDs, example, ibuprofen. There has been some studies which show that ibuprofen may increase the risk of COVID-19, but we do not have any definite reports of this. So we still currently use NSAIDs on an outpatient department, and in inpatients, we tend to use narcotic analgesics. Cough medications like guaifenesin is commonly used, and we also tend to provide patients with restricted fluid supply. I have mentioned restricted over here because it's extremely important that our patient do not go to dehydration. At the same time, we should not provide the patient with excessive amount of fluid because it can result in pleural edema and it can exacerbate the hypoxia and shortness of breath. And finally, it is extremely important that the patients receive sufficient amount of rest. Now, some of the potential treatment options for COVID-19 that is available in the market is hydroxychloroquine. We know that there has been reports that hydroxychloroquine is found to be very beneficial, and there was a sudden increase in the sale of hydroxychloroquine, and India was one of the highest exporters of hydroxychloroquine. But according to the Solidarity trial, there was no significant difference in the primary endpoint of the 28-day mortality rate. Patients who received hydroxychloroquine they had a 25.7% mortality rate in comparison to patients who received usual care who had a mortality rate of 23.5%. Those who are interested can look into the Solidarity trial for hydroxychloroquine. The inference from this trial is that hydroxychloroquine is more likely to cause harm than good, but it may be useful in severe infection. Another commonly used medication is dexamethasone. As per the recovery trial, patients who received dexamethasone, they had a decrease in the mortality rate by 1/3 in ventilated patients and by 1/5 in patients who received oxygen. So there is some positive results associated with dexamethasone, but we know that long-term use of dexamethasone or any steroids for the matter is associated with immunosuppression. So steroids, they are only used in patients who have acute exacerbation of their respiratory illness. Other experimental agents that is used for the management of COVID-19 is antiviral drugs like lopinavir and ritonavir, but there is no evidence of benefit. Similarly, ivermectin is an anthelmintic agent which has shown some benefit in inhibiting the replication of the virus in vitro, but there is actually no evidence of benefit when we conducted the ICON trial. Another agent is remdesivir, and this drug has the most promising data, but again, we use this medication only in case of severe infection. Now, as per the ACTT trial, it was found that the patients who received remdesivir have it shortened the duration of hospitalization. So these patients had a recovery time of 11 days in comparison to patients who received normal care, and they were having a duration of hospitalization of 15 days. So basically, it helps to shorten the recovery time, but we have to keep in mind that remdesivir is not given to outpatient patients, and it is only used in hospitals and as a part of clinical trial. Some of the other drugs that is under investigation is convalescent plasma. Convalescent plasma is basically the plasma of patient who has recovered following COVID-19 infection. In addition to that, we have use of immunoglobulins like phlebo-gamma and interleukin-6 inhibitors. Now, our hope is on vaccines. Trials are going on, and as of mid-December 2020, we have 57 vaccine candidates who are in research, particularly saying there is around 40 vaccines that is in phase 1 to phase 2 trials, and 17 vaccines are in phase 2 to phase 3 trials. The most successful vaccines that is currently with information available is the Pfizer-BioNTech and Moderna. And we have received positive results from interim analysis of their phase 3 vaccine trials. In Malaysia, we are planning to have our first batch of the Pfizer-BioNTech vaccine available by February. But we don't know the long-term immunity and efficacy of vaccines. We know that the virus has been mutating. There are several strains of the virus. It is still mutating. We have cases where patients who are infected are getting reinfected. So I think it is too early to tell right now the efficacy of vaccines. We still have to wait and see how it goes. Now, some of the challenges that is faced by clinical pharmacists, particularly in Malaysia, is, I think it's global, but one of them is spread of fake information. There has been some information in the media that drinking alcohol will help to eradicate the virus. There has been some information like bleach will help to eradicate the virus. In Malaysia, we have heard that Chinese sub-communities have the highest chance of developing the severe strain of the infection, whereas others are resistant to the virus. So there is several miscommunication and misinformation that is available in the media. And as pharmacists, it is our responsibility to get rid of these fake information. Secondly, there is insufficient drug supply. We know that when the pandemic struck Malaysia as well as globally, people were in panic and they were hoarding medicines, and there was a shortage of even essential medications like acetaminophen and medical devices. You might have come across photos where in supermarkets, there is absence of toilet paper, there is absence of mask. I went to a pharmacy, there was no mask at all. So there was a depletion of the basic commodities. And even in newspaper, there was a report that when hydroxychloroquine was found to be beneficial, a doctor ordered around 200 prescriptions of hydroxychloroquine to patient, even when we did not have the safety and efficacy results. So because of that, patients who actually needed hydroxychloroquine, for example, those with rheumatoid arthritis or lupus, they did not receive the medication for their treatment. Those are patients who require hydroxychloroquine on a day-to-day basis. And we are simply hoarding up the medication, expecting that it would be beneficial. So I just want to highlight that there was a deficiency of drug supply. And the third challenge we faced is that there is inability to provide pharmaceutical care services. In Malaysia, there was a reprioritization of health care resources, especially in the beginning of the pandemic. And because there was a limited supply of essential items like personal protective equipment. So clinical pharmacy services, they were limited to screening prescriptions and doing basic clinical pharmacy activities, and all the other special valued care activities that we provide were kept on hold. And finally, we had a restricted access to hospitals and medicines. In Malaysia, with the movement control order, it was extremely difficult for patients to come to hospitals and purchase their medicines. I just want to highlight to the attendees over here that the scenario in Malaysia is slightly different to other countries. We do not have a dispensing separation. Patients come generally to hospitals to collect medicines, and utilization of community pharmacy is generally very low in Malaysia, especially because medicines are cheaper in hospitals in comparison to community pharmacy. So implementation of movement control order, it restricted the patients their ability to come to hospitals and pick up the necessary medications. Now, moving on to the role of clinical pharmacists. Following the pandemic, there has been an expansion in the duty and of clinical pharmacists, in addition to the regular ward-based pharmacy activities which we provide on a regular basis. Most of the clinical pharmacists might be familiar with the terms I have written over here. The first one is medication reconciliation, and it is one of the most important intervention for every patient entering the hospital, because it helps to ensure medication safety and effectiveness on arrival, during hospitalization, as well as at discharge. Now, medication reconciliation is extremely important in the current situation. We know that healthcare providers, particularly physicians, are very busy with treating the pandemic, and they focus on what is in front of them. And they might not take much consideration of medications which the patients have been using chronically. But it still matters, especially if these medications are critical medications that is needed for the management of their condition. So we should, as pharmacists, make sure that we do not cause harm to the patient due to unintentional omission. Next one is participation in ward rounds and providing pharmaceutical care services to patients. We know that as clinical pharmacists, we are a critical part of the multidisciplinary team, and we should participate in ward rounds, and we should provide pharmaceutical care services to ensure medication safety and transfer of care. Now, in COVID-19 pandemic, we have to pay specific attention to antibiotic usage, because most of these patients, they have associated infections, and we tend to overuse antibiotics. I've come across cases where patients are receiving up to 5 antibiotics to treat community-acquired pneumonia. So we have to pay attention to the number of antibiotic. We have to see the spectrum of activity and see whether the antibiotic is particularly useful for the patient. Similarly, we have to switch to oral forms of antibiotic when it is safe to do so. The term we use over here is IVOST. It means IV to oral switch therapy. Similarly, we have to check the patient's liver and renal function, and we have to recommend dosage adjustment for those who require the dosage adjustment. We have to monitor adverse drug reactions and conduct pharmacovigilance. So we know that patients are going to require, and they will be given a combination of medications, and susceptible patients, they will come up with adverse events. For example, one of the most commonly used combination is hydroxychloroquine with azithromycin, and we know that this combination can lead to cardiomyopathy in susceptible patients and increase the risk of cardiac failure. Similarly, dexamethasone, we know that overuse of dexamethasone can lead to decreased glucose tolerance, and it can result in hyperglycemia. And this is particularly of concern in patients who have diabetes. In addition to this, some of the other services that was provided among Malaysian clinical pharmacists is remote screening of medication charts to identify MRPs. This is a common thing, but why I'm telling over here is that, as I mentioned in the beginning of the pandemic, clinical pharmacy services across hospitals in Malaysia were actually partially or completely withdrawn. And clinical pharmacists were asked to resign to their supply focused roles and not to do any medication review. However, most of the clinical pharmacists in Malaysia, they still try to access the patient charts, and they still identified the medication related problems, and they arrange video calls with physicians to get updates on the clinical status of the patients in the ward. And in addition to this, they also recorded videos of themselves giving instruction on how to use medical devices. And they tried to give it to the nurses so that the nurses can show it to the patients as a part of the medication counseling. So these are some of the innovative steps that were taken by the Malaysian clinical pharmacists, even though they were asked not to provide these services because of lack of PEP. So the next role and responsibility of clinical pharmacists is medication supply request. It is a general responsibility. We have to make sure that all patients in their wards receive a sufficient amount of medicines when they need them. In the outpatient department, because of the COVID-19, we make sure that the patients receive a 30-day supply of medications. But we have to be mindful about the drug interactions that the patients might face within the use of these medications. As I mentioned earlier, many of these patients have associated infection and they require antibiotics like azithromycin. Being a macrolide, it has a high potential for causing interaction. So we need to be extremely cautious of the drug-drug interaction among the medication. In addition to the medication, we have to make sure that there is sufficient availability and we have to ensure the quality of PPE, mask, as well as other medication devices. The next responsibility is discharge planning. As a part of the hospital regulation, we have implemented that patients should leave their ward within an hour of their decision to discharge, and we want them to leave the hospital within 2 hours of this decision. They should not stay in the hospital for more than 2 hours. So this means that the discharge letters will be prepared in a hurry and it might contain medication errors. So we have a great responsibility over here. We have to go through the medication discharge chart and see if all the medications are correct and appropriate for the patient. The next responsibility is providing patient counseling to patients for their discharge medications to ensure medication adherence. And finally, we have drug information services available in Malaysia, and they have a very huge responsibility. They have the responsibility of educating the public regarding COVID-19. For example, they have to educate the patients regarding social distancing, maintaining 2 meters apart from each other, appropriate use of masks. Still, we do not use the mask properly. We tend to keep the mask below our nose. We tend to use the white side of the mask outwards. So we have to counsel the patient regarding appropriate use of mask. We have to counsel them regarding proper hand hygiene with regard to use of soap and water, and they need to wash it for 20 seconds, about changing clothes immediately after coming from outside and washing themselves before being in contact with any other members of the household. And similarly, as I mentioned earlier, we have to advise patient on misinformation that has been spreading through social media. In addition to this, as pharmacists, we can also assist the hospitals in developing treatment protocols. There has been several studies on COVID-19 that is conducted across the globe, which says that this drug is beneficial than the other, this medication is much better, but there is lack of poor evidence. So we need to constantly update the current evidence of pharmacological management on COVID-19. Other value-added services that were provided in Malaysia were delivery of medications through postage services. So any patient with high risk of developing COVID-19, we should instruct these patients not to come to the hospitals. And we can actually deliver all these medications through postage services. In Malaysia, we have a postage service called Pos Laju. It is not free, but it helps to deliver all the medications at a very low cost. The next value added services was providing patient counseling as well as other pharmaceutical services to vulnerable patients, especially patients who are elderly and others who have chronic conditions through internet. We also had mobile health units, so it can reduce the influx of patients to the hospitals. We also had a pharmacy appointment system where the patient has to book in advance when they will come and the time they want to come. So it can reduce their waiting time in the hospital. As you can see in the figure, we have drive-through pharmacy. Now, this is almost similar to that of McDonald's and Subway. So patients can actually arrange for the medicine to pick up. The pharmacist would arrange the medicines. They would call the patient and give a registration number and time when they have to come to the driveway. And as soon as they reach, the pharmacist will go and supply the medication. And we have a locker for you pharmacy, as you can see over here. In this system, the pharmacist will fill up the prescriptions and they will keep inside the locker. The patients would receive a registration number, a code. The patient just have to go and enter it onto the locker and the medication will be, they can take out the medication from the locker. In addition to this, we have integrated medication dispensing system. Now, as a part of this system, the patient does not have to go to the hospital where they were initially reviewed. They can actually get their medication from any hospital across the country. We also have to provide psychological counseling. We know that there is social isolation. We are not seeing anyone. We are staying at home. So studies have found that there is high incidence of depression and anxiety among patients as well as the general public. So we can actually identify who is suffering from anxiety, and if they actually require counseling, we can direct them to a psychologist, or if medication is required, we can advise them to go and see a psychiatrist. In addition to that, pharmacists have been trained in administering COVID testing, and they have been widely involved as frontline providers. We have started trials on vaccines in Malaysia, and pharmacists are widely involved as clinical coordinators for trials. And once the vaccines are available in the market, we can also counsel and educate the pharmacists on proper administration of vaccines, and this would be useful for us. Now, with regard to the conclusion, I would like to say that although we are actively involved in delivering patient-centered care, pharmacists are often overlooked and our role is endangered. So we are recognized as frontline workers by other healthcare professionals, but this knowledge is not translated to the public or the news media or the politicians. If you see news, no one is giving value to us. No one is congratulating us for the services we are providing. So this could be because we are not proving ourselves and showing the impact of our activities to the public. So if we do not demonstrate our need in direct patient care, we may lose our opportunity to gain public support. So two major steps that I can tell you guys to ensure our position is firstly, we need to document and showcase the innovations that we have developed during this pandemic. And these innovations can be used to raise awareness and demonstrate the role of pharmacists to the general public as well as other stakeholders. Secondly, we need to evaluate what clinical pharmacy services we have provided during the pandemic and obtain feedback from physicians and other healthcare providers. The feedback from these members can increase our demand for clinical pharmacy services and even prevent future withdrawal of clinical pharmacy services, especially in case of future pandemics. So that's all from me. These are some of the references that were used in the preparation of the presentation. And I hope the presentation was useful to all the attendees over here. I just would like to directly address any students and clinical pharmacists, especially the early career ones who are there among the attendees. I just want to tell that we have a lot of potential within ourselves, but we need to demonstrate that to the physicians, healthcare providers, and general public. I was working as a clinical pharmacist, and it's a common thing that we tell that clinicians do not pay attention to us, or clinicians do not take into consideration what we are saying. That is normal. We need to be active. We need to be proactive. We need to talk to the clinicians, not to the PGs. We have to directly involve with the healthcare providers. We need to talk with the patients, interact with them. It might take time to see our role in the public, but we have to still maintain our activities, and we have to be active in the global scenario. And we have to bring up research ideas to improve our roles and responsibilities. So with that, I hope there is improvement in clinical pharmacy in India as well as globally. Thank you all.
Thank you, Dr. Menon.
Thank you, sir.
Yeah, you have motivated all the students and attendees who are attending this meeting. And actually, in Rajasthan, we have very few number of colleges who are running PharmD courses. We are only 4 in Rajasthan. Rather, we have a number of colleges which are imparting this PharmD course in the South India.
Yeah.
So you have fulfilled some of our dreams that we have to introduce this education to this state, especially Rajasthan. In this platform of APP 9th Annual Convention, we have made it. Only 4 colleges are running presently PharmD, and they may also require your motivation. You are saying rightly that we have to proactively, significantly depict ourselves to the physicians and patients. So we must be ready to serve, first of all. Thank you. And I thank Dr. Ramesh, who has proactively started reciting the profile of a student. That is also a kind of pride to you, profile being recited by the teacher. So congratulations and thank you very much for your presentation. And now I would like to call upon
Professor Jaybalan.
Yes,
I want to say a few comments. So I would like to congratulate Dr. Vinita, as
Thank you,
I
sir.
said earlier during your PhD Viva. So I was impressed there and today also. And that's why my idea was to include Professor M. Ramesh and you in our APP community and clinical pharmacy division to serve on a professional platform, because we are lacking the persons in pharmacy practice area in the association, and my idea is to improve it. And I think by involvement of both of you, so it will grow at a higher stage. So thank you very much for your input.
Thank you for having me, sir. Thank you, Ramesh, sir.
Yeah, thank you, Vinita. It was a very nice presentation. It was highly, you had done a very good job, and it was the presentation was highly informative, and it has thrown a lot of lights to the working pharmacist, and also as well as for the budding pharmacist. I suppose certainly they will take all those points which you have discussed during your presentations, and that will help them in taking the courage and moving forward. And this.
Hope so. Yeah. Thank you.
And Dr. Rajiv Dahiya, I should thank you, and as well as Dr. Jayabalan for giving me an opportunity to be a part of this whole to chair this session and being a part of this 9th Annual International Convention. And certainly, as you suggested, and there is a long way to go, and we should take a lot of initiatives to uplift the profession of pharmacy practice in India. And I hope this APP may be one of the forum where it can play a greater role in further taking up this pharmacy practice in our country. So whatever we certainly we hope that we would give our best efforts, and with your support and the organization support, and we will try our level best to uplift and contribute to this division.
Yeah, handled
Thank
by
you
a
for that.
leader. JSS Aher is taking the lead role in bringing in the clinical studies in India. So we are very happy to have you in this forum, sir. And now we have one more speaker to present his presentation, Dr. Vetriselvan Subramanian, sir. Sir, please start your presentation so that we can complete it.
Good afternoon to all of you.
Yes,
Thanks
sir.
for inviting me as a speaker of this virtual conference. Sincere thanks to Dr. Raju Diya, APP President, and the organizing committee members. Without further delay, I will go to my session. Can you see the slides?
No.
Yes, sir.
Yeah.
It's coming now.
Now it's coming.
Make it a slideshow, sir.
Today, my topic is translational animal research in drug development. So, how the animal research will transform to the clinical development and drug developments. In this topic, I am going to focus on what is animal research, why animal research is important to scientific developments, and how animal research useful to develop the drug and drug discovery and drug development of vaccines. So how to this animal research, how useful for vaccine developments. See, these are the areas I'm going to focus this presentation. According to Koop, he's the former US Surgeon General, he stated that all history of medical research, scientists solving medical problems, developing advanced techniques and treatments, curing of disease, all by using animals in research. So without animal research, none of the medical advancement in the world. Through the animal testing, prove the safety and efficacy of new treatment for both humans and animals. So without animal tests, we cannot prove the safety of the drugs. Virtually every major medical advancement through the animal research, especially among the laboratory animals or experimental animals, mice is more often used animal rather than other animals. So this is an introduction about which animals are is a very useful for the advanced research, especially with mice is one of the more common animal because the genetically have more similarity to humans. Animal research has played a vital part in nearly every medical advancement over the last decades. Nearly every Nobel laureates in physiology or medicines since 1901 has relied on animal data of their research. So when they're showing their research outcomes, without animal research, they cannot show any evidence. So this is one of the important criteria to prove their studies. We share a 95% of our genes with the mice, making them an effective model for the human bodies. So why they taking off animal study for the drug development? Animals and humans are very similar, physiologically very similar. We have the same organ systems, performing the same task in more or less the same way. Moreover, the same physiological conditions. Animals also suffer from similar disease like humans, including of cancers, tuberculosis, flu, and asthma. So that we go for animal studies before going to the clinical trials. Research on animals leading to the development of highly active antiretroviral therapies. So the antiretroviral therapies through the animal research only developed in 30 years ago. Since 3 decades before they developed antiretroviral therapy through the animal studies. While Fleming discovered penicillin drug without using of animals, the scientists shared the Nobel Prize with Florey and Chainhu. Those were tested on the mice, those were tested on the animal tests. Then only they discovered the full approval, the discovery of penicillin drugs with full approval could be used to fight infections inside the body. It's the first penicillin in the world. So all the drug developments and new drug discovery through the animal research is one of the main criteria. See, animal research has developed and helped modern vaccines, including polio vaccines, tuberculosis vaccines, meningitis, and recently papilloma virus, which is very important for cancer, particularly for the cervical cancer. Especially in Asian populations, most of the females, the more common cancer is a cervical cancer. So this cancer vaccines also developed through the animal studies. So the development of tamoxifen in animals led to 30% of fall in death rates from breast cancer in 1990s. So the new drugs development is animal is one of the main role to develop the new drugs. 450,000 people in UK suffering from type 1 diabetes rely on insulin. So, which was developed through the experiments in rabbits and dogs. In UK, they reported that how they developed insulin drugs through the animal tests, particularly with the rabbit animal experiments and dogs tests. So, through the animal experiments only, they developed vaccines and insulins. Through the animal research, smallpox has been eradicated from the earth. So the smallpox is one of the threats to the worldwide, is one of the major issues, but now fully eradicated from the earth. So that how the animal research is very important to develop new drugs and new vaccines. Animals in laboratories around the world have played an invaluable role in helping us to understand the disease. Why we need to go for animal studies is to helping us understand the disease conditions and what we need to do to treat it or cure it. So then only we can understand the exact mechanism of physiology. Based on that, we can go for further investigations. How animals have helped. Improved understanding of how genetic differences play a role in the development of the disease. Once we are doing with the animal studies, then only can understand what is the difference between the humans and animals, the genetically what are the differences are there. Based on that, we can go for further investigations. Then we can get a conclusions. The discovery of vaccines, the development of medical device, also not only for the vaccine developments or not only the drug developments, also is very useful for the medical device developments, especially for the incubators. Or so these are medical advancement through the animal studies only. Even though ventilators nowadays is very important for COVID-19 situations, most of the advanced ventilators machines, the device through the animal studies only, is not directly from the laboratories, directly from the companies. See, look at the slide. The clinical trials of a new medicine assess its efficacy and side effects can only be conducted in humans after new medicine has passed animal safety tests. Once animal safety test done, then only they are allowed to go for human trials or human study. So the new drugs must be shown to be safe and efficacy or effective in human subjects before going to the FDA approval. So it's one of the primary criteria, or it's one of the important criteria to get a approval from the FDA. So animal studies help establish boundaries for the safe use of the treatment when human testing or clinical trials. The animals models on a potential drug substances are required to follow good laboratory practice guidelines. So we must follow proper guidelines, national guidelines and central guidelines. Based on that, we used to conduct the studies. Finally, we have to submit the reports, the respective authorities. So during the studies, we must follow the regulations. The important role of GLP governs laboratory facilities, personnel, the productive equipments, and the usage of laboratory animals and operating procedure. These all are components of the GLP monitoring. These are the factors. Suppose if you are not following the proper laboratory facilities or proper room conditions during the investigation or during the your reports, maybe when they verify, they reject our research report. So according to the guidelines, we should keep our room temperature or facility or operating systems. Compliance with the GLP requires procedures and documentation of training, study schedules, process, and status reports, which are submitted to the facility management and included to the final study report to the Food and Drug Administrations. From the lab to FDA, you must show all the evidences. Without evidences, they reject our proposals. See, the preclinical studies data are gathered to reach the goal of potential therapeutic effects, and a reasonable safety index is very important. So we must show the safety of the drug substance or test substance, and the drug sponsors must be notify the FDA of its intent to test the potential new drug in humans. So they have to explain scientifically whether our drug substance is a safety or not. Then only they can allow to go for further steps or further approval. Shouldn't use humans to human disease study. It is not always possible. We are not directly testing on humans. It's not ethical also. By law, all drugs and medicines are required to be tested on animals and other in vitro tests to protect the human rights of people who volunteer for drug trials. Once studied with the animal studies, our study is a safety or effective for the animals, then only we can go for human trials. So this is the ethically, this is the one of the rule. We should not directly go for human trials. So one of the important criteria for this, for the clinical trials. An important part of most animal studies is examination of tissues and organs post-mortem. Most of the studies when we are doing with animals, we are doing tissue tests, organ tests, which would not be possible for human volunteers. So that animal study how is important, because we are not directly inject any drug to the tissues or organ, is not at all possible for the humans. So that we must go with animal studies. Once safety is there, then only they can allow for they can allow to go for human trials. Animals can be bred and kept in controlled conditions and fed standard diets to reduce the experimental variables. So better we should follow the guidelines. If you are not follow the guidelines, some deviation will be there. If too much deviations are the result is not appropriate. Sometimes by end of the verifications or end of the submissions, they reject our results. So we should go for proper procedures. Why do we still need to use animal in research? What is the importance? Why we need to still keeping of animal research? Advanced scientific understanding. Without animal study, we cannot understand the physiology or disease conditions. And solving medical problems. Protect the safety of people and animals. These are the important criteria, or these are the important components why we need to have animal studies. Animals are used when there is a need to find out what happens in the whole living body. So this not at all possible with the humans. Directly, we cannot inject any new drug substance into the humans, which is far more complex than some of its parts, especially with the humans. It is very difficult and in many cases simply not yet possible to develop non-animal methods. Is a non-animal model is very difficult to understand the disease mechanisms and the physiology of body. So that's what the animal studies is one of the very important criteria to develop the drug development, to develop the drugs. Obviously, is there any difference between humans and animals? Definitely the difference will be there. But under the skin, the biology of humans and other animals, particularly the mammals, is remarkably similar, same physiology, especially with mice. So more than 90% physiology is normal, and more than 99% is genetically have similar to humans. So this is very important. That's what nowadays most of the researchers, especially with the cancer researchers, they used to go for mice animal with altered genetically engineering mice, altered genetically engineering mice. So these all why the laboratory animals are very important. See, we have the same organs controlled by the same nerves and hormones as many other species. Where there are differences, researchers know about them. Such differences can actually help scientific understanding of your particular problems. Once we do with the animal models, then only can understand the exact problems or how to rectify or how to overcome the issues. Then only we come to get a conclusion. Without animal studies, it's very difficult to get a conclusion. Or exact mechanism is very difficult to identify the exact mechanisms. See, many animals suffer quite naturally from the same disease as humans. Can be used study those diseases. Usually, the animals and humans, most of the diseases are similar, especially cancer, tuberculosis, and other respiratory diseases. In other cases, researchers can use an animal model of a disease which is close to human conditions. So also, this study is very useful to understand the physiology of animals. Once they understand the physiology of animals, they can easily correlate to the human beings. Most medicines or treatment are only available as a result of extensive research. So the research is very extensive, takes a longer time. Within a short period, very difficult to develop any drugs. Development and testing of drugs taking many years, often decades, taking a longer time. The study of animals throughout this process gives valuable insights to explore several ideas and several ideas, then explore some knowledge on your particular studies, how the healthy and diseased body works. So this is the reasons animal studies is very important in research. This means why are animals used in scientific research? Still, most of the researchers and research laboratories, still they're using laboratory animals, especially the mice animal, genetically modifying animals like mice. Particularly these animals they're using for cancer research can make them even more like humans, similar to the humans, and even more useful in understanding the disease. Animals are also used to see whether new chemicals have harmful effects, such as finding out whether a chemical causes cancer or harms the unborn fetus. Also, the animal studies is very important to identify the cancer disease conditions also to identify the effect of pregnancy. So these are the research animals is very important. More than 90% of medical advance through the animal research. In 2019, the Great Britain, the Great Britain, approximately more than 60% of animal procedures they have done for the scientific research purpose only. More than 60,000 research they have done. See, it means without without reasons they cannot go for animal studies. To understand the whole body and the way different parts such as the heart and lungs and increase nervous system is interconnected. So you see that normally our human body is interconnected with all parts and all organs. It is vital study to whole body. So we cannot directly administer any substance to the humans. So that we initially go with the preclinical studies, then only we start human trials. It is not yet possible to create artificial animal models. So with the living animals only is it possible to conduct the research. Other tests like estimate effective doses and find out the best way, not only to develop the disease, to develop the drug developments and vaccines. Also, our drugs are very useful to identify the routes of administration, which route of administration is appropriate for the patient. Also, the formulation of drugs, which dosage form is better for the patients. So these all by animal research only. So these are the factors we are doing animal research. Laboratory mice are more often used animals compared to other animals, make up more than 90% of the total number of animals used. You know, approximately is out of the research, approximately 90% with rats and mice. The remain animals only 10% they are using. Likely is dogs, cat, rabbits. This is only 10%, but most of the research doing with mice and rats. The statistics stated that in 2019, 3.4 million procedures were carried out in Great Britain in the living animal, only for the scientific research. Around half of all procedures were experimental procedures, whilst the other half were the creations and the breeding of genetically altered. It means around 1.5 millions of animals they used only for the experimental procedures and genetically altered purpose. The genetically altered purpose, especially for the cancer research. And majority of procedures used with mice compared to other animals. Approximately 93% of animals they used with mice. Over half-life of experimental procedures were for the purpose of basic research. Most of the research, most of the animals they used for basic research. Some animals, some percentage they used for advanced research. So ultimately, all research involved animal research. Animals, all research involved animals. See how are animals used in medical research? Drug discovery and development. Animals are used in early stages of research to investigate how organisms and disease behave. For the development aspects, animals, the new drug, medicines, vaccines are tested in animals to ensure they are safe or effective for use of the humans. So this is the purpose we are doing for the animals, especially in the drug developments. Research involving animals such as mice with share 99% of our DNA can help scientists find out the new ways to fight disease in humans. So this is one of the key point why we need to have animal study. So this is the drug discovery and development process. These are the stages. Initially, they synthesize the chemicals. Before synthesize the chemicals, we need to make a team. We need to create a team or form a team of researchers. Then they go for synthesis of new drug substances or new the chemical substance. Then they identify the safety or efficacy. Then they go for formulations, animal tests. Finally, they report to the investigational new drug with the FDA. Then only they allowed to go for clinical trials. So these are the stages, drug discovery and developments. I'm not much explain about drug discovery and developments. Just I am focusing on why animals used in the research. So the initial stage of the pipeline focus on the discovery and validations of newly identified disease-associated drug targets. So these animal studies, why we use for animal, main reason is to target the disease conditions. It means once you administer the drug, our target in particular disease only. So how the drug substance work on the particular site of area. So based on that, we can go for further investigations. The preclinical models thus provide a bridge to the clinics, to connect to the clinical trials, and are requesting part of the drug development pipelines. So the preclinical is one of the key area, one of the platform to translate or to bridging to the clinical trials. So these are the references why the animals are very important. Just I referenced, I cited here. So cancer, especially for the cancer drug development. So applications of mice models in cancer drug developments. The development of novel treatment strategies in oncology requires preclinical studies in mice cancer models to identify the validate novel cancer drivers and therapeutic target, how our drug is targeting to the disease site. So based on that, we can determine what is the effect of drugs, what is the efficacy of drugs. Also, we need to look pharmacokinetics, how the drugs moves to the body. So what is the absorption of our new drug substance? What about our distributions, like how much drugs are distributed to the extracellular, how much drugs distribute into the intracellular. So these all kinds of things by the animal studies we can easily identify. Finally, we can get a result of the dynamic response. So what effects produce, whether it's a therapeutic effects or toxic effects, or there is no response. So these all research is very important to develop the cancer drugs. So when promising preclinical research are obtained, the tolerability and anti-cancer efficacy of these drugs are evaluated in human patients in phase 1 to phase 3 clinical trials. So these all animal studies is very important and valuable to give ideas to go for clinical trials, especially phase 1 to phase 3. A proportion of patients will show poor response due to the intrinsic or acquired resistance. Also, we can identify from the participants here, especially with the animal models, we can easily identify, is there any intrinsic activity, especially for the agonist activity, or is there any antagonist activity in any particular receptor sites. So we can easily identify from the animals, which may be studied mechanistically in preclinical mice models to identify the response biomarkers. Also, we can check for the blood tests like biomarkers or immunological markers. Also, we can, by the laboratory test, we can find out what biomarkers are elevated or maintain the normal range. So easily we can identify through the animal studies. The close alignment of mice studies and human clinical trials will lead to better patient stratifications. So easily we can identify the proper concept or the proper mechanism, how the drug is act on the human's body. So this one comes through the animal studies only. We cannot directly finding from the patients. identification of novel biomarkers and development of optimal combination therapies, and culminating in improved cancer patient care. So also we need to check with some combination therapy with the animal models. Then we can find out whether the cancer markers reduced or increased, or there is no change. These all by the animal research only. Initially what we are doing, initially we induce the cancers, then we are giving some drug substance, then we are analyzing whether the cancer markers reduced or increased, or there is no change. Based on that, there are many advanced research they're doing with animal models. According to the European Association of Experimental Animals, she has looked at the key stages in the development of coronavirus vaccines. In the European associations also stated that the animal model is very important to develop the vaccines, especially the nowadays is one of the global issues of coronavirus. So research for vaccines starts in labs, where scientists try to understand the nature of coronavirus and the way it attacks the body. So it's very difficult to with the human beings. Some mechanisms are very difficult. It means some studies are very difficult with the humans. So why even though the vaccines of corona, many animals they're using for to develop the to identify the to develop the corona vaccines purpose, they're using many animals. I will show you the further slides. They will often study animals that experience the disease in similar ways to humans, often at a molecular level. Even though the molecular level, some the similarity is same. The current models used as to answer questions about COVID-19, especially with ferrets, is one of the animals. Hamsters, pigs, mice, and monkeys. These are the animals nowadays ongoing research with the COVID-19 vaccines. The next step is testing the vaccines on animal to make sure the vaccine is safe or effective. This is a legal requirement set out by health regulators. Mice and monkeys are usually used in this phase. Usually, these two animals are very common for the initially to develop the vaccines. Some argue that to speed up the process of vaccines developments, the vaccine developments, animal testing stage can be avoided and testing can begin straight away in humans. However, this is fraught with the dangers. We cannot go directly to the humans. We should go with the preclinical models. Then only can get the proper mechanism, or then only we can understand the proper mechanism of drug actions. After animal testing, the vaccine is ready for clinical trials, which means testing in human volunteers. And if these are successful, the new vaccines will be approved by the national and international regulators. So after testing animal only, they're checking with the humans. Then they will get a approval from the national and international regulators. So this is the animal. Currently, they're using this animal. In the search for vaccines and the treatment, especially this is for the mice. See, in the search for vaccines and treatment for COVID-19, animals can play an essential role in both basic and advanced studies. Mice play essential role in both vaccines and drug development for COVID-19. Nowadays in UK, majority of researchers, they're using mice animals to develop the COVID vaccines. Many research companies also, they're doing with the mice animals. Ordinary mice are not susceptible to the disease conditions. So the ordinary mice, they're not using, so that the scientists breed genetically altered mice. Genetically altered mice they're using to make them susceptible to COVID-19. So these are the animals models they're using for to develop the vaccines. The second animal is hamster, is one of the laboratory animals being used to study the effect of the coronavirus, as these animals are a good model to study the effect of the virus on the lungs, especially in the lung system. This animal is very useful to identify the exact mechanism of coronavirus. So the hamsters are being used to study the effects of COVID-19, as they can display similar symptoms to human when infected. So after infected, once infected with the COVID-19, the same symptoms appear from the hamsters, so that easily we can correlate to the humans. This similarity makes also them a good model for studying transmissions, the effect of the virus, particularly on the lungs. This animal, especially the lungs, the sensitivity is very high compared to other animals. So that this animal is very useful respiratory. This animal is very useful for respiratory disease conditions, to testing on the particular disease conditions, any respiratory disease conditions. Then monkeys. Monkeys also is a very common animal to develop the vaccines, have similar immune system to humans, and currently being used in many coronavirus studies across the world. The most common type of monkey used in the rhesus macaque, but scientists are also conducting studies using African greens and marmosets. So these animals currently they're using to develop the corona drugs and investigating the vaccines, corona vaccines. Ferrets is one of the important animal have been used to study COVID-19 because this animal show similar symptoms of humans, especially the lungs physiology, especially with the lung physiology. So they are often used studies of influenza for similar reasons. Already these animals and all, they are using influenza studies, influenza research. So this animal is very helpful to identify the exact mechanism of the lung disease, especially with coronavirus. Another animal is a pigs. This is often used as researchers. This animal used for COVID-19 testing to help scientists better understand how the drug is safe and effective for the new vaccines, and therefore whether it is suitable for human trials or not. Also, apart from that, the vaccines, these animals are also being used to test the effectiveness of new types of ventilators. So device also is very important, when the development of new devices, especially some ventilators. So this type of animal very useful to identify or to develop the advanced ventilator machines, advanced ventilator device. So before they are tested on humans. So these all importance of animal research, why we are doing animal research for the medical advancement. These are the notable views. According to the British Heart Foundations, most of the researchers, the research outcomes, especially for the heart disease, especially heart disease, in that aortic valve replacement, these procedures and all they developed through the animal studies only. And cancer research UK. The cancer research, most of the cancer research, especially for the breast cancer, all drugs developed through the animal studies only. Also, the scientist, the Albert Sabin, who developed the polio vaccines, he said that without animal studies, he cannot develop any vaccines. So that how the animal study is important to develop the vaccines and drugs. With this, I concluded that all studies is very important and essential to develop vaccines and drugs. In that, animal studies also is very important to identify the human physiology and to identify or to understand the mechanism of disease. Thank you all.
Thank you, sir. Thanks for your presentation, sir. And
Thank
now
you, sir.
I request respected chairpersons of this session, Professor Subhash Chandra Dinda and Professor M. Ramesh sir and Professor Anju Goyal ma'am, to ask the questions and submit the end note of for the session. Please.
Thank you, sir.
Yes, sir. Please carry on.
So it's a nice presentation. First of all, I congratulate Dr. Beatrice Selvan, and very, very informative presentation, which is a need of the present day. Because after post-COVID, I think India has realized that where we have to move, particularly when government is giving stress on the research and academics to strengthen the research and academics in India, particularly national education policy they emphasize. So this trans we have to develop strength or emphasize on the translational research, and particularly using these animal models when we are particularly COVID and all, not only COVID, we are now stressing on the COVID, but there are a lot of areas we have to develop, particularly anti-cancer and AIDS, and there are so many complications already existing with us. But without animal study, developing well model to study a particular drug or dosage forms, and this gives too much information, and I think our researchers today, it goes a good message that the researcher what they have to concentrate, where they have to concentrate to strengthen their research and give good output, which can give the societal benefit to the community. So productive research is very important, and where Dr. Selvan has emphasized the different models, very important models to study, including COVID. So I think this is a very good presentation, and not only he, all the presenters, all the resource persons today, they were resourceful. They have a nice presentation. They presented very well, even though people's how they are deteriorating the ethics now because of mushrooming of the institutions and the universities. Everywhere stress, and people are diverting from the ethics. Everywhere management stressing, you give 2-3 papers, yearly 5 papers. Is it task? Are they going through the reality? All the vice chancellors, what they think, I don't know. Only all are competing to get the NAAC accreditation A grade, NAAC accreditation A grade. Only paper you give. Is it a way? And all are manipulating, and plagiarism is high in case of India. So we have to think about our moral values. We have to think ourselves that what we are able to do, and without the diverting from the ethics, we have to emphasize ourselves, concentrate ourselves, and we should not deviate from our goal. And we have to stress, particularly research, we should not compromise. We should not compromise. And now I think government also realized where we have to develop. They are only stressing on the sports and all. Now they realized where they have to invest, because we are back in research. Why the development of the vaccines are lagging behind? Because we are not well developed lab and our scientists. So they are not emphasizing how much they are paying to a scientist to work 30,000, 40,000 per month. And you see how, which way the other professionals they are earning. So that's why people are discouraged to concentrate on the research. So I think this is this time we have to emphasize on, realize where we are. We have to realize that in which way we are progressing, our country is progressing, and where we have to strengthen. University should realize, the university management also should realize that yes, what can be done, how we can strengthen our research facilities, how we can motivate the researchers to stay with us in long time and give a good outcome that can be like a translational research, and it can be utilized for the benefit of the society. Very good way, Dr. All the today's presenters, they have emphasized in like Kamal Dua, Dr. Kamini, and Beatrice Selvan, and Dr. Binita. So all I have listened to them, each individuals. Very nice presentation and nice research they are emphasizing on. This is the productive research we can utilize. We can utilized for the benefit of the society. That is important. In which way we will give benefit to the community. So I think all are presented well, and also our delegates and all participants. It gives a good message to the participants. Thanks to all the participants for being with us and sharing the knowledge from these type of scientists who were with us and share their knowledge and spare their valuable time with us. Thank you all. Thank you. I realize that this is a very fruitful, there will be very fruitful outcome, and this seminar is a success. Thank you. Thank you.
Thank you.
Thank you. Thank you, Professor Subhash Chandra Dinda. And we like to hear from Professor Anju Goyal, ma'am.
Yeah, thank you all the eminent speakers for their wonderful lectures on the different field of pharmacy. Like today you have included all the pharmacognosy, professionalism, ethics, pharmacy practice, clinical practice, SFC, that means pharmaceutical chemistry. So today I think it's the rainbow of lectures of different fields. And definitely students will get participants, students, researchers and faculty will would definitely get the extract of different lectures. I think so. It is a grand success, I think, for Jia Balan sir and organizing committee, and of course Rajiv Daya sir. So really hats off to energy and stamina of the convener and organizing secretary and organizing committee for availability from morning to night. Matlab, I'm observing this that from 7:45 in morning to now it's up to 5 p.m., 6 p.m. They are continuously here on the screen. So hats off to energy and stamina to all of you. And I'm very thankful to Dr. Rajiv Daya sir, founder president of APP, and Dr. Jia Balan sir, convener of this 9th APP annual conference, international annual convention, for giving me this opportunity to chair this wonderful session. Thank you, Dr. Vinita, Dr. Kamini, Dr. Vetri Silvan, and Dr. Rohit Kumar. So thanks for your efforts, and especially thank you, Dr. Sunita Daya madam, Yogendra sir. Thank you.
Yeah, thank you very much, ma'am. And we like to hear from Dr. Sunita Daya a few words about the lectures.
Thank you, sir. Just like yesterday, it was a very nice experience to listen to various areas, especially areas from the pharmacy practice and clinical practices. Usually we are more concerned with the areas of the pharmaceutical sciences, but it was today very nice to hear from all the speakers from the different areas of the pharmacy practice. I hope this session have been fruitful to the participants and faculty as well, like from the area that we are not working with. Thank you to all the eminent speakers for sparing their valuable time and sharing their research and their thoughts and their scientific input into this conference and making this conference a grand success. And we would like to have more participation in our future events. Moreover, just like Anju madam mentioned, that the untiring efforts of Professor Jai Balan sir and his teams, hats off to you, sir, that continuously your enthusiasm is not affecting with so much of activities, and it motivates to us also. Thank you so much.
Thank you, ma'am. Thank you. Actually, this is the first lesson that we learned today to not to have any online convention on Sundays because the electricity board and telecom board do not understand that the people may work on Sundays too. So there are so many difficulties in connecting with the internets and electricity. We are keep on calling the officials of telecom and electricity board as it has gone. So this is the thing happens on Sunday, and the people are taking a rest. Sometimes it is also a wrong decision to live stream the events through this thing, YouTube or Facebook. then that it would be very easy for people to watch YouTube whenever they like to, and they just skip the live activities. So,
but Prof, nobody will feel bad. All know about their state.
But the
situation of the electricity.
But the speakers are very nicely done the job, and they justified it, and they have given. That is what I wanted to have a plethora of thoughts come in one panel, one platform, so that we can hear everything that we can do as a pharmacist. So. The paradigm shift now happens in Rajasthan after hearing the lecture from Dr. Vinita. We are looking into the domain of the practice nowadays. We started very late, actually, in practice side. B-Pharmacy is there in everywhere. We are, of course, we are having one of the oldest institution in Rajasthan, but for PharmD, we are lacking behind that we have to accept. And we needed somebody has to talk about the add-on services that you have mentioned one by one in the hospital. How can we take lead role in the patient care and safety? So these things will definitely induce some of the minds of the students to have at least go for some post-baccalaureate education in PharmD after their B-Pharmacy. So that is what we needed to have at present time as far as Rajasthan is concerned. But we are doing this APP 9th annual convention in global level, and we are having leaders from across the globe to talk, to deliberate. So this is very happy moment for me as well as my team and the host institution to have all of you in one platform and seeing all the faces from different countries and different places. Somebody from Japan joins and leaves, and somebody from South Africa joins and leaves, and somebody from West Indies, somebody from. So every for every person for their effort. Yesterday, one senior Professor Patak woke up at 3 a.m. and talked for 1 hour, and then he went back to sleep. So this is something that happens with the love and affection that they have with Professor Daya and the Association of Pharmacy Professionals. And we hats off and to their commitment. We thank their commitment. And with this fifth session, we are completing all the.
Prof, I have the questions. You not ask me.
Plenary lectures. And so we give this opportunity to all the panelists and the chairpersons now, Dr. Dr. M. Ramesh has joined. Sir, if you have any questions to be asked to the speakers, sir, this is yours time. Okay. Over to you, sir.
Okay. I just have a couple of questions to Vinita. Vinita, I just want to know that, as we all know that there is a lot of myths about COVID-19 disease and as well as about its management. How best we as a pharmacist can diffuse the communications of these myths and also the inappropriate use of medications in COVID-19 patients?
So with regard to the misinformation that is spreading regarding COVID-19, the only thing we can do is educate the patients. We have to counsel the patients repeatedly, like what is COVID, what is the method of transmission. You have to wear proper mask. There are anti-mask communities worldwide. We have to educate them that mask is important. You are not resistant to the infection. You have to wear the mask. Similarly, they say that there is homeopathic medications. There is that helps to prevent you from developing the infection. There is no medication available that will prevent the disease. There are medications that are available in Ayurveda and homeopathy that will boost our immunity. So we have to educate the patients clearly. What are the medications available? What are the supplements available? What is the mode of transmission? And what are the precautions and preventary methods that you have to implement to prevent developing the disease? The only thing we can do is proper care and hygiene. prevention is what we can do at the moment. With regards to cure, there is nothing available in the market that can cure the disease. There are some in vitro studies that shows that this medication is beneficial, but we also have to look at the side effect profile. We have to see the pharmacovigilance aspect of every medication. We know that hydroxychloroquine, it has some benefit, but it is associated with more side effects. It can cause cardiac arrhythmia. For a drug that improves your improves your chance of recovery by 1%, would you take that medication when it has a 99% of causing cardiac arrhythmia in patients who are susceptible? So we have to look at the safety and efficacy profile, and the only thing we can do is, as a pharmacist, we have to constantly update the guidelines. We have to educate the physicians. I wouldn't say educate, we have to update the physicians, like these are the guidelines that are available. We have to do systematic analysis and meta-analysis and show them that these are the guidelines. This is poor evidence. It has high amount of heterogeneity. You can't rely on this. You have to rely on these articles which have more evidence, which have less bias. So from a care perspective, I think we have to update the treatment guidelines and provide doctors with updated evidence.
Thank you, Vinita.
Thank you, sir.
And my last question is, so what we have learned so far, or what should we learn as a pharmacist after being, part of this healthcare team in the management of COVID-19 pandemic?
Okay, as I mentioned during the presentation, we need to learn that we have to be proactive. If we just take it as a job, go to the pharmacy, just be there for ward rounds and don't do anything and come back, it is not a clinical pharmacist's responsibility. We have to be there for pre-rounds. We have to interact with the patient. We have to collect the details from them. We have to see if there are any MRPs or DRPs, identify if there is any medication errors, communicate pharmacovigilance. If there is any ADRs, we have to communicate it to the responsible authorities. We have to talk with the doctors. We have to talk with the PGs and the nurses, as well as other healthcare professionals. We have to actively interact with them. I think being proactive is the most important need right now. We have to be proactive. If we are not proactive, we will be left behind. And if we are left behind, what is the role of clinical pharmacy? What is the role of all those before us who have tried to implement this profession in India and globally? If we as future pharmacists, if students as future pharmacists do not take active responsibility, I don't think healthcare providers and public will understand the importance of clinical pharmacy and pharmacy practice. So I think the need of the hour is to be proactive, get involved in research, try to understand what is happening globally and try to implement that in our society. Yeah, thank you.
Vinita, so that's all from my side, and I'm not sure whether are there any questions from the participants. If any questions from participants, I think you can take it forward.
No, sir, we didn't have any question in the chat box as well as in question answer panel. So,
Professor Jayapalan?
Professor Jayapalan, sir, can you hear me?
Yes, sir. Yes, sir.
I wanted to add some more points. Just now, Dr. Sunita told for the awareness and prevention and all. Just we have to educate our students. In hospital side, our role is limited. In hospital side, our role is limited, pharmacist role. So we have to educate our students. The students will educate to the society. They will get some more awareness in the, if the students will get more awareness, maybe they can educate to their family members. So this is the way we can move forward, or we can give, effective the awareness. We can go this way also can. This is what I feel that, because in hospital is a very limited our role.
Yes, sir. Thank you, sir. Thank you.
Sir, may I add one point?
Yeah,
yes, sir, please.
Yeah, particularly what I observed for the PharmD students, and if you want to develop a clinical pharmacy in India, this I hope that experiential learning, which is lagging behind in India, it is very, very important. And if we developed that particular area and practice and clerkship, particularly during clerkship for the students, what I observed near abroad, they are emphasizing on this particular aspect, and pharmacists show their potential in this particular area and can improve the therapy, improve the drug therapy. So I want to add, this should be emphasized, particularly for the PharmD students. Those who are running the PharmD courses, they should give stress, according to my point of view. Thank you.
Just one add one comment to this. I think know that if you look at the curriculum of the PharmD, already the experiential education is in the curriculum.
Yeah, but not practiced well.
It is only thing the institutions should ensure that the students are properly trained, because they do 6 months of clerkship and 1 year of internship where they get a complete experiential education. In fact, at JSS here, Mysore, even at the early stage of their course and the program, we would train them through the simulated settings. For example, how to do the counseling, how to conduct a patient medication history, all this we have how to prepare an IV admixture. We train the student through simulated settings before they get it into the real practice settings. So that would give a complete understanding and the basic skills that is required to practice in the hospitals. So all institutions should impart this kind of training and education to students. Certainly students will have not only the interest but also they will be more confident, because they've already done it through the simulated settings. They will be more confident to play their role in the hospital when they do the experiential educations. So when during the experiential educations, the preceptors who are there in the hospital, they should train the students, with a very close supervision. If you look at in India, there are hardly very few institutions, their staff will spend their time in the hospitals. Otherwise, they spend all their time in the wards. Then how we can train our students? It is not possible. So even the staff has to take, they will be more proactive, and they, the steps should be initiated to ensure that staff spend a lot of time in the hospitals. Only then the staff can train the students. If the staff are not spending their time in the hospital, how can we expect the students will get a good training? So it is all this. It is all this experiential education is already in the part of curriculum. It is only thing is how best as a faculty and as an institution, okay, what responsibilities we have, how to what extent we are, administering the proper training to the student is a most important thing. That would, finally design what would be the competencies which the students are developing, okay, when they graduate.
Yes, sir.
I just want to add to what Dr. Ramesh and Dr. Subhas have mentioned. When I came to Monash, something that was different is the presence of virtually integrated modules. So over here, in addition to just teaching theoretical aspects to the students, they are also involved in providing active workshops. So theory is there, but at the same time, they are involved in getting students involved in workshops where they are being exposed to live cases. And before going to hospitals, they are being taught how to identify MRP. It's already there in JSS curriculum, but I think many institutions do not provide this kind of training to students, like how to identify MRPs, how to effectively counsel the patients. And as Ramesh sir mentioned, our students have abundant knowledge. Indian students are very intelligent. They have high amount of knowledge, but they are very passive when it comes to clinical settings. They do not implement it. They are afraid to talk with doctors. they tend to, even if they go to ward rounds, they will be at the very back. So the professor is there, the assistant professor is there, the PG is there, the interns are there. Then comes the pharmacist. The pharmacist at the very back, they do not talk with the professors. And as soon as the ward round is done, they will go back to the department and they'll just sit there and talk. So that should be avoided. The students should be taught to be in the pharmacy. There should be some preceptors to monitor them, what they are doing in the pharmacy. So I think from this time itself, when they are doing clinical placement itself, they should be taught the importance. They should know that they are important in that setting. There is a wide responsibility and role that is available for them in that setting. So if we provide them with that kind of perception, maybe they will improve their activity and responsibilities towards the patient. Yeah, thank you.
Thank you. Thank you very much. And
one question, sir. There is a query in chat box by Muhammad Adil. As pharmacy profession is mostly profit driven, mainly towards factories and mainly compromise ethics. So does both goes side by side? I think this question is for Dr. Rohit Kumar for professionalism and ethics.
Is left, I think so.
Okay.
no problem. I will forward the question to the resource person and I'll get the answer from him. Thank you, ma'am. Thank you for forwarding the question.
Chair, may I get the chance to ask some questions?
Yeah, please, sir.
So my question is for Dr. Kamini. So you are working on cyclic peptides. Are you synthesizing cyclic peptide or isolating?
Hello, can you hear me?
Yeah.
Yeah.
Okay. It's not cyclic peptides. It was so that was a peptide that I purchased. I didn't synthesize that. That was it's a linear peptide. So it was tetrapeptide. So it was from the subunit of insulin from the beta chain. So it was from the 15 to 18 residues on that subunit. And then the nonapeptide was, I think, 15 to 23 located on that, the beta chain, the subunit. So it wasn't cyclic peptide. It was just a linear peptide of a length of 4, 8, and 9.
So have you done any protection or deprotection of free amino or carboxyl group?
No, but with this particular instance, we didn't synthesize it. So, but in the lab,
I am asking as a whole, as per your laboratory point of view. It is not for your research, but your supervisor. Okay. Are they doing work on cyclic peptides?
I can find out about the cyclic peptides, but I was learning some solid phase peptide synthesis.
Okay, so because I'm working on solution phase peptide synthesis. That's why I was interested in.
Okay, we do a bit of solid phase here.
Okay, thank you. So my another question is for Dr. Subramaniam.
Thank you.
He explained about use of animals. So using the animals and using the softwares, there are two scenarios, and particularly killing extra animals, and that issue was there. So we moved towards using the software. So how these both situations will impact on research?
Good question, sir. We can minimize the number of animals, but we cannot totally stop animal research. We can minimize the number of animals, but we cannot stop.
You can continue.
Yeah, even though what you are telling, like bioinformatics, like with animal model, we are checking with the computer modeling or other things. Just we can understand the mechanism, but exact mechanisms with living animals only, not from the computer model or like some other technology. So better we have to go with the animal model, but we can minimize the animals. That's all. Even in the guidelines, in India also, I had the last meeting. I had a discussion with in the Indian experimental animal director. So he also telling that so we can reduce the animals, no issues, but we cannot totally stop the animal research.
Because as in India, when I was the principal, so there was directions at UG level, you can't use animals, you have to use the software. So it was like that. So is that was
It's appropriate.
You agree with that?
Yeah, I agree this, because undergraduate level we can go with some animal model or some software. Some software we can show, like we can show the bioassay tests. Also we can show some animal models, some the software. But when they go for PG, postgraduate and research, they must have some animal studies. Otherwise they cannot understand the exact mechanism of their research. That's what, even though either any area like a pharmaceutics or chemistry or any area, they finally they can come to animal study. Then only they can prove whether the study so and so or what is the effect of their drug substance. So,
Hello. Okay. Thank you.
Yeah, thank you, sir. Thank
Hello.
you.
I can add one point.
Yeah, please.
Actually, the animal research is prohibited or restricted, not for study or general study like B Pharm level, even though PG level also, they have to go for model and computer simulated programs. But for the research purpose, particularly if it is so important and approved by the institutional ethical committee, you can go for, I think.
Yes, sir. I agree with the proper guidelines.
Yeah.
Yeah.
Thank you.
You can sacrifice only animals when there is a so desire and for the benefit of the social
society, when the benefit to transform the clinical trials.
So one last question to Dr. Vinitha. As we were discussing about those CLACSIP modules and those, I think those problems can be better solved by MOUs between the universities. So maybe in a foreign university, a better healthcare plan is there, or healthcare training is there, that some of the students can opt from India, and then they can develop same model in India, and by that way we can be in the better healthcare chain. I think it is workable, and we are doing in our medical school and nursing school. They did collaboration and then they developed here.
Yes, sir, I agree with that because Monash University in Malaysia is collaborating with Monash University, Australia. So most of the curriculum and the method of teaching and the method of providing pharmaceutical care services is derived from Australia. What they are doing over there is exactly what we are trying to implement for our students in the hospital setting. So I totally agree with you. If we do collaborate with countries, especially universities where the pharmaceutical care services are well established, and we try to bring that to our country, then there is high chances that we can develop pharmacy practice as an established method of pharmacy in India.
Okay. Can you comment lastly on COVID vaccines, like in Russia developed and UK developed? So how it is working?
Well, when I went through literature regarding COVID vaccine, what I found was we cannot be sure whether it would be efficacious, because there are people who got infected with COVID-19 and they got reinfected with COVID-19 again. So the principle of the vaccine is we are going to administer the virus, the RNA, to the patient. They are supposed to develop antibodies to the virus. So if the patient is getting reinfected, that means that the strain is undergoing some kind of mutation, or the vaccine is not able to produce, the antibody is not able to produce the beneficial effect in the patient's body. So based upon the literature, they are saying that they are not sure whether the vaccine is going to be efficacious. But if you look at the Moderna, the treatment schedule, the vaccine will be administered on day 0 and it will be re-administered after 21 days. So we are not sure if we follow that treatment plan, whether the vaccination will be successful, but we can only hope so that it can be. But so far, based upon the phase 3 results, the companies have released that the data is found to be very beneficial.
Thank you. So I think with this, I am thankful to all the speakers and the scientific judges. It was a good discussion, good and informative, and it will be beneficial for all the attendees. So over to Jabalan, sir.
Yes, sir. Thank you, speakers, and thank you, chairs. And I would like to thank one by one by mentioning their name, because Dr. Kamini has presented for his first presentation, and she is waiting for a long time with us and hearing all the lectures. Thank you, ma'am. Your presentation on chromatography for the purification of biologics was a wonderful presentation. And Dr. Vettiselvan has given the use of animals in the experimentation. So thank you very much, sir. Your patience. We have tested your patience.
Sorry for the inconvenience. Sorry for the inconvenience. My net connection is also interrupted.
Dr. Vetri, I was not thinking yours will be possible, but but thank you.
Thank you, doctor.
We thank Dr. Kamal Dua and Dr. Rohit Kumar Verma, who are not here by this time, but their presentation was very contented and it was good. And now, Dr. Vinita, thank you. We may be disturbing you in the future also for our students. So thank you all for your wonderful, amazing presentations. And we especially thank Professor Subhash Chandra Dinda, sir, for his mind-blowing thoughts on the education, industry, and practice. Sir, thank you for your thoughts and thank you for your speech. And Dr. M. Ramesh, I have several times watched him through Zoom meetings. This time I'm sitting in the other side. Otherwise, he'll be moderating. I'll be hearing Dr. Ramesh always. Thank you, sir. Thanks for coming into this APP 9th Annual Convention, and thanks for your services. And from my house, I have another chair who is Dr. Anju Goel from Rajasthan. He's a very good friend of mine, and thank you, ma'am, for being with us. And in the meanwhile, she served a cup of tea through the mobile. Thank you. Thank you for your tea also. And thank you, my dear colleagues, for supporting in this event. And with this, we are completing the 5th session of plenary lecture. And in another 15 minutes, by 3 o'clock, we'll be starting e-poster presentations for the students, those who have submitted abstracts. And the email has been dispatched to all the participants as well as evaluators. And one Google form has been sent to all the evaluators for put the marks for the presentations. And it has been divided into 5 groups. And I have broken the Zoom meeting room into 5 different rooms, so that in different rooms, different participants can join separately, and the proceedings will happen. Within 1 hour, we have to complete the proceedings, and from 4, we will be joining again for the valedictory, where the award and yes, sir.
Sir, one of my scholar, I think he registered for e-poster. Mr. Bibhas Chandra Mahanta. I think he didn't get any link or something. He was telling me.
Sir, I have sent it. Okay. While you are talking this session, I have completed, and that he has been informed.
Okay, thank you. Thank you.
Thank you very much, sir. Thank you. And thanks all of you for supporting us in this APP 9th Annual Convention. Thank you.
See all of you in the valedictory session. Please
see you all. Please, all of you are welcome there. Thank you.
So we'll join with same link, sir. We'll join with same link.
Yes, sir.
No, valedictory different link is there.
Okay.
Same link. Oh, I heard we can join with family. You can join with family, but the link is separate. I will send it to you, sir.
Okay.
There is a link of valedictory session separate.
Yeah.
So you can.
Thank you, sir. With the permission, I end up the session. Thank you.
Thank you for having us.